computational models. And ideally, tomorrow you hear about this from Jason, how might we consider how these lists are used in the world where AI is all over the place and chat, GTP, and all these buzzwords that people are saying. We as stewards need to make the data that's used in those approaches useful and well curated. So finally, that gets me to the roadmap to make these results useful to the community and really ultimately enable discovery. Because what we're all doing here is trying to improve biology and human health, right? That's the end. That's what's out there right at the end of the day. So the way that we thought about these two perspectives, and this is what I really like. Each of our resources come at this problem from two different perspectives. And I'm going to walk you through that real quick. So the Knowledge Portal Network, what we've done for years is think about summary representations of human genetics. We started back in 2015 with the first version of the Type 2 Diabetes Knowledge Portal, which had 9 GWAS data sets across 25 traits, right? But the goal there was not to be like a repository, even though we had to ingest the data at that time. We really made it such that you can summarize the genetic evidence for your favorite gene, region, or variant, right? So you can see it, it's accessible to you, you can interpret if you're a non-expert. And that's really been our bread and butter. We try to think about that from all sorts of perspectives. Additionally, what we try and think about is, by bringing in those summary representations, what can you do for the field to perform integrated analysis? What methods can you run to produce better results, like meta-analyses, computing credible sets, incorporating genetic annotations, SNP to variant linkages, all sorts of things that help you hone in on what is the variant, what tissue, what gene, and so on and so forth. But do that in the environment of summary representations. And that's like the curation is the key part here, right? So it's an integrated platform. And then finally, sort of expanding this beyond diabetes, we expand this to an entire community, thinking about folks in disease spaces, trying to understand what genes or variants are associated with the disease, and then what path to follow, provide genetic support, if you will, right? So the whole hallmark is there. We work with communities to define what they want to see. It's a flexible platform and it gives on-demand knowledge. So that's the perspective that we bring, right? So the curation, the presentation of summary representations. The GWAS catalog complements this activity so beautifully, and they've sort of like set the tone for the entire field of human genetics, in my opinion, is they are a repository for this. They're the place where you put the data. And back in 2020, I think I got that right, Laura, they said, hey, GWAS data are all over the place. We want to develop standards. They held a workshop, which was part of the spirit by which we're holding this one, to define standards for the community, bring experts from different sectors together and say, what are the challenges? What needs to be done? What's the minimum viable product you need to get data incorporated into one repository where it can be accessible to all? And that really was something that they do really well, right? They develop those standards, then make them accessible. They make it super easy for you to share summary statistics. And now what they develop community standards. They're sort of the benchmark. They're a resource for access, and they're a resource for on-demand data. So the complement there is natural for us. So how we come at this perspective is today, we want to think about everything from the ingest to the presentation and to the use, right, over the course of the day. So with that, I'm going to quickly walk us through the landscape. I'm a little behind on time, but that's okay. I will go fast. So day one is really about the landscape, right? So day one is about, things that people and perspective. So we're going to kick off with Eric Fauman from Pfizer. He's going to talk about his perspective for many years about thinking the causal gene. So really set the tone for us, thinking about as a consumer of these lists and someone in industry saying like, okay, human genetics field, what are you doing to help me? And how can I use that? So that's his perspective that he'll give us. And then Yakov from Open Targets, who really is one of our partners in organizing this, will think about as a consumer of these for another resource, what's valuable, what matters. So that'll be this morning's session. And then Maria will really hit the stage and really talk about her, I would say, 2-year-ish effort of curating and really, she's done an incredible job of thinking about what's the landscape, what data have been produced, curating all the literature, and for the 150 lists that we've already curated, she's done them all. So it's pretty amazing. She has this interesting perspective that she'll give that will really set the tone for how we approach these challenges. Then we'll talk some approaches. Well, we're going to work through 3 people who have worked with us with various approaches for effector gene lists. First, Brent from McGill. He'll talk about his perspective and how he thinks about effector gene lists. Then Cassie, who we've worked with also, who will also talk about her perspective from a disease state as well. And then we will have Adam Butterworth, who will join us from across the pond to talk about his perspective. So those will be perspectives from different disease areas and different ways of laying evidence sources. Then we'll have an open discussion. We'll sort of breathe, reflect on what we've learned. It's going to be a lot of talks this morning. And then we'll have a nice total flip. So Marina from ClinGen is talking about how ClinGen thinks about how they represent gene predictions and effector lists within their resource, which will give us a totally different perspective from the rare disease side of the world and how they've done it. I think that would really help to guide our discussion. So then we'll adjourn. And then you can go home and you can synthesize your thoughts, rest, or work the rest of your day, because many of us will be halfway through a day. But day 2 is ready to work, right? So for that, we're going to have a lot more interactive session day 2. Laura's going to kick us off, though, from GWAS's perspective, GWAS Catalog's perspective on how they've done this so far, and think about how they did for GWAS standards. Someone might need to mute, unless there's a question. Thank you. Right. And then we will have Maria come back to talk about presentation of these. How are we getting them accessible now? What are we doing now? What is the resource? How does it exist now? And then Simon from Open Targets will do the same about how they are presenting the gold standard list they've produced. So that'll be a nice perspective there. So then we'll have sort of convert to open discussion. We'll have an open discussions about the pros and cons, the key features, who should we target, who's our end user. We'll talk about what matters in terms of these lists for contributors, people who are generating the list and contributing them to this resource that we will develop standards for. We'll talk about as consumers, how are we going to use them? What's the important pain points in someone using them as a person in industry, a person in biology, and so on and so forth. And then we're gonna have this really cool readout of the surveys that many people, we had, just to step back for a second, we have about 200 people interested in joining this workshop, which gives you a sense of what matters in the community. We had about 50 people fill out the survey. We have about, how many registered attendees for this? We capped it at around 90? Yeah. So there's 90 of you that over the course of the next 2 days are going to be participating. We had to turn people away, so it's really our job to share this with the world, which is kind of cool. Not that we turn people away, but that we get to share this with the world. So what did the community say? What are the gaps? And so you'll hear that survey result. And then Jason's gonna pivot and have us thinking about, okay, well, now that we have all these, how might you integrate them? How might you evaluate them? How can they be useful for other approaches in the world of large language models, in the world of knowledge graphs, and all these things that we as folks building resources are thinking about, but trying to figure out how they all connect to the actual biology and data we're generating. So that's going to be Jason's remit. And then finally, we'll present a straw man. So we'll pull this all together, and Laura and Maria are going to walk you through, like, what is yeah, I have fun with these figure generated. They're really fun. So a straw man for what might be a way to develop these standards. And we're going to talk about it. We're going to poke holes in it, and we're going to make it more structured over the course of time. So that's what we'll get to work. We'll have breakout sessions for about 40 minutes, guided by questions that we'll provide you. We want readouts. So we want the leaders of the sessions and the note takers to really help us read out and discuss together. So each group will break out and come back together. We'll go through this tomorrow, obviously. And then this will help us develop a roadmap and really talk through some of the challenges. And our job as organizers will be to synthesize all this and bring it back to the community. And hopefully when we're done, we'll have a set of standards, we'll have a resource, and we will have a community of stewards. And that's really what I'm hoping to get out of this. And I really want you all to feel engaged and like you all matter to making this work. So I'm so glad you're here. And, you know, I have one anecdote, but I think I'm gonna save that for tomorrow. Because I've been thinking about this for a while in terms of, you know, how we present this to the community. And I have an anecdote from Anna Gloin and how she used what we've currently done, which would be valuable. But I think I'm gonna save that for tomorrow. With that, I'm gonna stop. I'm gonna turn it over to Eric for our first talk of the day. So Eric, I'm gonna stop sharing and you get to kick us off.
Else is sharing. There we go.
There you are. I can see you.
Okay. Let me switch to full screen. Hold on. Good. Yes.
I love the backdrop. That's cute. I didn't see that at first. I thought you were in an aquarium, but now I feel it's a little avatar-y too.
The theme is Pokemon. Right?
Oh, okay. All right.
Okay.
Yeah, definitely.
Okay. Are you seeing? You can see my slide now, yes?
Yes.
Good. Okay, so first of all, thanks for the invitation. This has kind of been an obsession of mine for the last 13 years, so I'm happy to be able to talk to you all about it. There are a lot of sort of stories or vignettes or sections in this, and so I think and we have a whole hour. So I think if people want to ask questions along the way, probably the most effective is if people put in the chat, and then Noel or Maria can read those out as we go. And I guess I will pause at the different sections. The section joins are pretty obvious, I think. But this figure, and yeah, I also have stuff in here about generative AI, and we get to that. But yeah, these figures here, I also had a lot of fun with. These are all from ChatGPT from DALL-E. And this figure really does represent, I think, part of my own adventure in getting through this. And so for folks that I haven't had an opportunity to meet, my training is not human genetics per se. My training was biochemistry, biophysics, and I did X-ray crystallography for PhD and postdoc. But structural biology of biochemistry related enzymes. And so this is sort of where my core is understanding proteins, enzymes at a molecular level. So my introduction to GWAS, and I've been at Pfizer now for 26 years. My introduction was when we had a collaboration with Nicole Soranzo to look at metabolite QTLs in the twins and Cora consortium. So this was my first introduction. I hadn't heard of a GWAS. I didn't know how they were done, but I started seeing these lists, just as Noel was talking about, lists of lead SNPs and their associations, and trying to make some sense of them. And what immediately was apparent to me was that a large number of them, when we're talking about metabolite QTLs, and we're going to get a lot more into this, when we're talking about metabolite QTLs, a lot of those can be understood in the context of sort of classic biochemistry, and that's what got me hooked. And again, 13 years ago, and I've sort of gone through there. So this sort of thing, now this particular Manhattan plot comes from actually the MetSim consortium, the MetSim cohort, and this is work that I've been doing with the folks at University of Michigan and in Finland. But so this is a GWAS of phenylalanine, and there's only one hit, and it's on chromosome 12. And as we're going to be talking about over and over again, the question is then, okay, who's that causal gene? And just to make sure we're all on the same page, I'm going to take a step back before we get back to that. And that is where I've spent a lot of time is looking at metabolite QTLs. And we're going to talk about the assumptions in trying to assign what the causal gene is for a metabolite QTL. As I was looking through this, what is a GWAS where you can really make the simplest of all assumptions, and that's the protein QTLs. And there are really now a lot of these, and I will show you just how many there are. And again, just to make sure that we're all on the same page, since I'm the first presenter here, right here, when I'm talking about a pQTL, right, this is a variant which has a significant association with circulating levels of a particular protein. So in this case, the protein is the IL-6 receptor. There's a very strong association for this that sits on chromosome 1 and happens to be within the gene that encodes the IL-6 receptor. And I think, especially for pQTLs, and you'll see later on, we have to be careful when we're talking about the gene and the gene product. And I think, because oftentimes we use the same symbol for both, but sometimes we're talking about the actual biological physical entity that goes out and does stuff in the cell, the gene product or the protein, and sometimes we're talking about the gene itself. So in this case, the variant that affects circulating levels of the IL-6 receptor happens to sit within the IL-6 gene. Now here I'm representing actually work that was part of the UK Biobank Protein Pharma Partners project. This is the Sun et al. Paper that came out last year, I believe. And so what I've done here is I've picked out a number of pQTLs, protein QTLs that sit on chromosome 1, where the gene encoding that protein is also on chromosome 1. And again, just to make sure we're all on the same page here, you can see that there's a signal for an association for carbonic anhydrase 6, the protein that happens to sit within the gene that encodes that protein. Skipping over, we've got this IL-6 that we talked about. There's also an association for nitogen 1, the protein nitogen 1 that happens to sit very close to the gene for NID1. And so I think in each of those cases, well, actually, we'll take a look. What we're building to is it's obviously very limited number of assumptions you have to make to say, maybe the gene that encodes that protein is the causal gene or the effector gene for that. Now in here, I am also showing there's an association for alkaline phosphatase, which happens to sit within the gene mTARC1, even though the ALPL gene is also on chromosome 1. Right? And so then the question is, well, it's very far away. What actually is the causal gene here? Is it this closest gene, mTARC1, or is it ALPL, which is the causal gene? So in fact, if you go across all the pQTLs that you can find, what you do is you take every pQTL where the gene encoding that protein happens to sit on the same chromosome as where you see that pQTL, and you just ask what the distance is, right? So at this point, not making any assumptions other than that, right, that gene might be the causal gene and it's on the same chromosome, but not making any assumptions about that distance. And if you just look at what the distance is across all of these, what you see is this very rapid drop off, right? So these are in units of 10,000 base pairs. So the most are within the gene or within 10,000 base pairs of the transcription start site, and then that drops off very rapidly. And then actually my colleague and I, Craig Hyde and I, wrote this up because what we actually observed was if you do this across everything, you actually see this odd distribution. And again, I don't have time to do this full justice. And so I encourage you to look at the paper if you're interested in it. But in this case, to be able to look at the full distribution, remember chromosome 1 is 250 million base pairs. And so in this first figure, I mean, I'm only going out to 250,000 base pairs. So this graph would have to be 1,000 times larger. So if instead we just take the log of the distances, so now 1 million base pairs is 6, right? And 10 million base pairs is 7. What you actually see are these two different humps when you look at all the distances. And again, that was the observation that we put into the Fauman and Hyde paper. I'm going to digress just a little bit because this was a lot of fun because I was obsessing about, okay, why would you see this distribution with these two humps like this? And after thinking about it all summer after the paper, I think I've got a solution that kind of makes sense here. And again, if you want to read more on this, this wasn't worthy of its own paper, but I did push this out to a LinkedIn article where I've got more of the details there if you want to take a look at this. I like this because it seems like a fun kind of thing that undergraduates could play with and show that this works. I call this, right, pin the regulatory element on the genome. So you just take 20,000 promoters and 200,000 enhancers, and you just randomly scatter them across the genome. You just pick random positions across the genome, and you place them there. And if you then just say, if you pick any random pair, so you've placed them now, and now you pick a promoter and any enhancer that happens to sit on that same chromosome, and you ask, what's the distance between them? It turns out, now here I am going out to 250 million base pairs. It turns out that, again, if you just pick two random points on a string, it's more likely those 2 points will be close to each other because there's more ways for 2 points to be close to each other on a string. There's only one way for those 2 points to be the total length of the string apart. And so you get this in regular distance space. But if you take the log transform of that, you put these into log distances, you get this peak out here. Just the way it appears. And this part we do talk about a bit in the Fauman and Hyde paper. But this is what you get if there's no specific relationship between the promoter and the enhancer, you just pick two random pieces. The insight I had here, and I think I hope over the next 2 days we'll talk a bit about these different ways of linking in enhancers and promoters. I happen to be a big fan of Jesse Eingritz's work with the ABC model. And so if you take that literally and you go back to the Fulco et al. Paper and you just pick out values that they have for the activity, the activity of these enhancers, and you randomly assign, so you've randomly scattered these enhancers across the genome, you randomly assign these activity values to those enhancers. And now, again, just in the simulation, you ask for every promoter, what is the ABC best enhancer near it? And now there's this trade-off because enhancers nearby might be weak, but but you penalize enhancers that are too far away. This is all part of the ABC model here, right? So the contact, the chances that two things will be in contact decreases rapidly as they go away. And so you have this interplay between how close the enhancer is to the promoter with the strength, the sort of cell-based, cell-specific strength of the enhancer. And so now you end up with this average distance of closer to, of more than, you're moving beyond the closest enhancers. You've got the closest enhancer, but oftentimes you have to go to one that's further away. And if you plot this now in regular distance, you get this sort of graph. And if you plot it in this log space, you get a peak out here around, between 10,000 and 100,000. Again, in real space, always closest is better. It's the trick of moving to log space is to better visualize it. But always remember that it is more of this rapid drop off. If you put those two curves together, again, from the simulation, you put these two curves together, you have this peak out here when there's no relationship between the enhancer and promoter. You have this other peak here if for every promoter you pick the ABC best. So this up here is purely from the simulation. Now, if you go to the GWAS catalog today, pick out every pQTL that's in the GWAS catalog. For every unique protein, you pick the strongest pQTL for that protein that lives on the same chromosome as the gene for that protein. And you get this plot down here. So this plot down here doesn't know anything about the ABC model or anything else. This is just straight from the GWAS catalog. And again, this is something that anybody can do. You take every pQTL for each protein, you take the strongest pQTL for that protein that lives on the same chromosome as the gene for that protein. That's this down here. And if you put them together, you get this. So I thought that was pretty cool. Obviously, it doesn't mean that the ABC model and the rest is true in every detail, but I think it does help us. Well, it certainly helps me, I think, understand sort of how these genetic or genomic effects are coming together. And again, when we talk about pQTLs, we often talk about trans pQTLs, where likely this is acting through a gene, not the one that encodes that protein. And these ones over here would be the cis pQTLs. And again, these drop off rapidly at about 1 million base pairs. And again, if you want to see more details on this, you go here. This was a bit of a digression. I will come back over and over again to that it is usually the closest gene. And I think you have to make a strong argument for why you think the closest gene is not the causal gene. And we'll show more about this. I was debating how much to put on this. Again, talking about ChatGPT. If I ask ChatGPT today if the closest gene is usually the causal gene, it tells me no. And then I have to show it all my papers. And then it says, oh, actually, maybe you're right. So it is not a one case at this point. And more specifically, if you go back to the pQTLs, so this is for the pQTLs, the cognate gene is the label I use for the gene that encodes the protein. For pQTLs especially, it is the cognate gene is also the closest gene 78% of the time. And that's true actually no matter what the classes of the lead SNP. Now we're going to talk about fine mapping and so forth when I think most of, maybe I'm going on. I think most of what's in the GWAS catalog is the lead SNP and not necessarily the fine mapped causal variant. And so for here, I'm going from what's listed as the lead SNP and the VEP effect of those. So certainly if the VEP effect of a variant is a missense in a particular gene, that gene is almost always the cognate gene, the causal gene. But even if it's just anywhere within the gene body, especially if it's within an exon, it's also going to be the causal gene, or even if it's within an intron. And again, or here, when I talk about closest gene, I almost always mean closest to anywhere within the gene body. And I've tweeted about this before. I guess I haven't written it up. But if you have a variant that sits within an intron of gene A, but is actually closer to the transcription start site of gene B, it's more likely to still be gene A. And so there's something special about the footprint of a gene. And wherever that variant sits within that gene is still more likely to be the causal gene, even if it's not close to the transcription start site. And then this last category here is even for the vast space between genes, even in those cases, whatever gene is closest to that variant is still going to more likely to be the causal gene. You've got a lot less confidence than if it's within the gene or if it's a coding variant. But in all of these cases, that's going to be the causal gene. I'm going to pause there for just a moment to see if there's any questions before I go back to the metabolite QTLs. Okay on any of that? Okay, I'm just going to dive on. I'm going to come back to the metabolite QTLs, which again was sort of my first love in this. Again, what I like about the protein QTLs is the only assumption you have to make is the central dogma that the gene encoding the protein is probably the causal gene. So for metabolite QTLs, you don't quite have that. But there are cases where there are very few genes that could be involved. And phenylalanine is one of those in this story around chromosome 12. And so some of the tools that I've used, again, what I brought into this, or what I realized coming into this, is that we do have now over 100 years of sort of classic biochemistry. And a lot of that is captured in a lot of nice databases. One that I really love is HMDB, which among other things, for all the thousands of metabolites they have in there, they have documented what are the proteins, enzymes that are known to act on that metabolite. So if you look up phenylalanine, what I'm showing here is this is the list that HMDB says these are genes these are genes, proteins, enzymes that act on phenylalanine. As you can see, only one of these happens to live on chromosome 12. And so if we assume that the causal gene for phenylalanine is a protein that acts on phenylalanine, HMDB is only giving us one option, which is this phenylalanine 4-hydroxylase. Another resource, which I think we will come back to, and I'll come back to in my talk, is mouse knockout studies. There are obviously other model systems, but MGI has made it really nice and easy to cross-reference human phenotypes and mouse phenotypes. In this case, you can say, what are all the genes that what are all the human genes that when the mouse ortholog is knocked out has an effect on phenylalanine levels, according to how they've annotated things. And then again, in this case, you can see that there are a lot of different genes that come up. Only one of these lives on chromosome 12. And then finally, again, was there a question?
There's a question.
Yeah, I have a question there. Do you mind if I stop here? Sorry, I was a little slow.
Yeah.
To the draw. I was thinking about what you're saying about, the work you've done. And I know I've also followed your blogs and your posts over time. And I was thinking about, and maybe you're going to talk about this, have you taken sort of like a look at the effector gene lists that have been out there, like the ones that say like, this is the causal gene, and actually looked back to say for like the diabetes list, the Mahajan and McCarthy one, which we'll talk about later, have you sort of said like, for the ones that they say are the causal, are they the closest gene? Like, have you validated that hypothesis with like effector gene lists that are out there?
So the Mahajan list is actually part of, well, okay, let's come back to that. Yes, let's come back.
Because I'm just curious, like, I'm almost thinking like, is proximity to the sentinel variant almost like a standard that we should have people apply in uploading their list as like a de facto thing? But anyways, go ahead.
Certainly one of the changes in the field that I've seen that I'm really happy about is I think it is impossible now to publish a method for assigning causal genes without comparing it to closest gene.
Yeah,
right?
okay.
I mean, that's got to be the baseline. And it's clear that we can do better than that, but you better show what happens if you just pick the closest gene. So, but the others, let's come back to those.
And Eric, can I ask a question?
Go ahead.
Eric, my brain was stuck on something you said a moment ago about that bimodal distribution of the distance for the PQTL.
Yes.
Have you done a similar analysis for EQTL?
The problem so in Fauman and Hyde, we do show that if you take the EQTL catalog, you get that first hump, but I only had access to data where the distance is less than 500 kb. So if I had a similar data set where people calculated every EQTL, right, genome wide for every transcript, then yeah, we should be able to do that. But I have not seen that data. Yeah. Right. I mean, for good reason, people generally only calculate cis EQTLs. But if somebody calculated genome wide, it wouldn't have to be genome wide. It would only have to be for the chromosome. Right? So for every transcript, do the whole chromosome that transcript is transcribed from, and then
Well, and allow for more than one QTL for that transcript on that chromosome.
right, and that's a whole other subtlety. So yes, everything that I've done is taking the strongest, right? So the strongest for each one. And I think yes, as soon as you go beyond that, other things happen. It'd be great to talk about that more. Because right, what I'm showing here about closest gene and so forth does begin to break down as you take right the second and third and fourth strongest signal, even at a locus, and see that in the metabolite QTLs. One of the reasons I haven't spent as much time with that is because there are lots of methods for identifying what are the secondary and tertiary signals. And especially for things like metabolite QTLs, if your primary signal is 10 to the minus 20,000, what kind of R squared do you need to ensure that a secondary signal is not at all connected to that P to the 10 to the minus 20,000 signal? So I think there's more work to be done there. But at the same time, there's good reason to believe that, right, if the primary signal maybe is because you're hitting the promoter and your secondary tertiary signal is because you're hitting some weak enhancer, those get further and further away. I think there is a lot more work to be done there. Okay,
Thank you.
let's wrap up this section. Because I think another theme that we will come back to, and certainly this is used in the in Anuba's gene list, is looking at rare disease, and this has worked really well for a lot of different GWAS. That if something is a rare disease gene, that is a good prior for it being a GWAS hit. In this case, if you say what are all the genes that are known to have, I guess, inborn in areas of metabolism that manifest on phenylalanine, there's only this one, phenylalanine hydroxylase. And now if you combine all of these, right, all these methods say, oh, there's only one gene that sits on chromosome 12, and it's PAH. And in fact, well, and in fact, that is exactly where that variant is, right? And so I think you would not have much argument to say, the variant that is affecting phenylalanine levels on chromosome 12 is probably acting through the effector gene is probably PAH. Just to wrap up this part, you can do this systematically across, again, everything in the GWAS catalog. Take every metabolite and say, what is the strongest signal for that metabolite on each chromosome? And then to each of these methods, the HMDB, the mouse knockout, the rare disease, to each of those methods, identify a single gene on that chromosome. And if so, how far is that gene from that metabolite? And this is a summary of that. The magenta triangles is the graph that we were showing before on PQTLs. But what I'm trying to show here is whether you're using the mouse knockout information or the rare disease information or the HMDB information, you get the exact same curve. Again, in the Fauman at Hyde, we describe that curve as coming from the Weibull distribution. We give the parameters in there. It is not an exponential decay. It actually has fewer parameters than that. And as I just showed, it derives nicely from the ABC model. But this exact same curve, I think, applies everywhere. And my argument for why should we care what happens with protein QTLs and metabolite QTLs when what we really care about is diabetes and IBD and so on. And my argument would be, how are those genes acting if not through proteins and metabolites? But we can come back to that. Okay, so I'm going to pause on this figure and walk through this because I spend a lot of time on it. Here's where, again, I got to play with our generative AI. I think because this makes several of the points that I think will come back. As I've thought about what are the types of information, the sources of information I use when I think about whether a gene protein is causal for a trait, I think they fall into these two categories. In the ProGem paper, we describe these as top-down and bottom-up. And in the POPs paper, L. Week's paper, they talk about similarity-based and locus-based. I think there's sort of they map here. In this context, I'm going to talk about biological features versus genomic features, or we can think about it as the fox in the nest. So the fox up here is the thing that's actually having biological activity. It's going out and it's doing things. It has partners, pathways, so on. It's what we know about what the fox does that lets us know whether the fox is involved in the phenotype that we care about. And wherever that fox is located in the forest, we would say, yes, that fox is probably involved in that phenotype. So again, things like what the pathway is and what kind of biochemistry it has and who its interaction partners are. So things that, and this is again why I'm emphasizing the gene product here. The Fox PAH is the gene product, and it's going out and doing things in the cell. So wherever that occurred in the genome, we would say, yeah, that probably could, that's likely to be involved in that phenotype. As opposed to the locus features, which in this case is the nest. PAH happens to sit at chromosome 12 at that position. A lot of the things that we look at are more of these locus features. How close is the nest to the lead SNP? Where does it sit within a TAD? Where does it sit in a high C kind of conformation? And so we would say whoever is sitting in that nest is likely to be causal because of where that nest is located relative to the genome signal. And I think especially then when those things come together and we say all these genomic features are pointing to that nest, and who do we find in that nest? We find this Fox that is involved in these features. We would say, yeah, that's highly likely. And one last thing I'll say about this, most of what I've done, again, I think because of my training or my interest, is trying to focus on the biological features to say, let's nail down that, yes, this is a gene like PAH, the phenylalanine hydroxylase, which everything that we know about its biochemistry and activity says that, yes, this is a gene that must be involved in that trait, or a protein that must be involved in that trait. If we can nail that down, we can then use that as a basis to assess all the locus features, things like EQTLs or high C or epigenetics and so forth. And maybe we will hear a bit about the locus to gene stuff from EBI. I think that's one of the things I liked about that paper, is that, right, I compile this list of here the genes that I think I can believe in from biology, and then from that we could run through and show that, you know, for the nth time, that EQTLs are not good at picking the causal gene for a variety of reasons, that that proximity and gene density are better features on the locus piece. So I will pause there for a moment, but I did hope or plan that this figure would be something that we might be able to come back to, I think, again, as we look at how different people are using different features.
Yeah, I think this is a great, like setup. I like Pokemon theme. But I do think it's the idea that there's biological features and genomic features, and those you'll see from actually Maria's talk, I think, as she sort of the different ways that those come to create a list, whether it's through a bioinformatic prediction or through a heuristic or what, you know, or straight up integrated method that does both. So I think this is.
I wasn't sure. Will Jason be talking about Pigeon tomorrow? No, okay.
We wanted him to, but we actually chose a different direction for him. So.
Okay, that's fine. And that is also on YouTube, I believe. But certainly the axes that he shows in Pigeon, I think, also then map to this, which is one of the reasons I love the Pigeon approach so much.
Definitely.
Okay. Let me do one more digression around you know, so I've sort of laid out all these assumptions of how one might pick a pQTL or assign a pQTL, and how one might assign a metabolite QTL. One nice piece of work, or at least that I'm excited about, that we worked on over the last couple of years, was you know, largely the assumptions in those two methods are independent of each other. And so what if we could compare them? So what if there's a locus where we have a metabolite QTL and we also have a protein QTL, and we can compare those and ask, do we get the same gene or we get a different gene? So to walk through one example, there's an enzyme, gamma glutamyl transferase. Among other things, it carries out this reaction, taking the cysteine glutathione mixed disulfide as substrate and creating that product. From work, both from the UK Biobank Olink work, and then also the MetSim work that I've been doing with Mike Benke and others at University of Michigan, we get a pQTL from the UK Biobank work. We have a metabolite QTL from the MetSim work. Josh Chu and my group ran through these co-localizations. So those two signals, you can see the locozoom plus, those two signals sit in exactly the same spot. So for that protein, we would say that the cognate gene for that protein is GGT1. For that enzymatic reaction, we would say the likely effector gene is also GGT1 because it encodes a protein that carries out that reaction. So in this case, we have from the pQTL, we say the likely causal gene is GGT1, and for metabolite QTL, we say it's also likely to be GGT1. And in fact, then you can run this across. So Josh did then do all the colocalizations of all the metabolite QTLs and all the protein QTLs to look for what are those cases where they line up. And then it was nice that he did that independent because I'd already assigned all the genes from the metabolite work. And then you can say, how often does the colocalized protein QTL agree with the gene or protein that I assigned from biochemistry? And you can see they line up where they agree 80% of the time, which is better than what you get if you just pick the closest gene. It's a lot better than what you get if you do colocalization with eQTL, which I took as nice confirmation that in general, the assumptions that we're making for pQTLs and for the metabolite QTLs are valid, I guess, because we're landing on the same gene. And we can take this one step further because these are enzymatic reactions. And as I showed, this metabolite is a substrate for this enzyme. And so you can say, if you had more of the enzyme, then you're likely to have less of the substrate because you have more enzyme, it's going to consume the substrate. So again, Josh did the analysis from the colocalization and then asking if the direction of effect were the same or different. Independent of that, and without looking at those results, I went through each of these pairs and I said, based on what I know about the biochemistry, would I expect that having more of that protein would make more of this or less of this, right? So if the metabolite is a product, then if you have more of the enzyme, you would expect to have more of the product. So sometimes they should be in the same direction, sometimes opposite direction. This will only be valid if what we happen to be measuring in circulation for the protein and the metabolite has any relevance to where the reaction is actually taking place. And it's only going to be true if I've picked the right protein right from the pQTL and from the metabolite QTL. And of course, enzymes are catalysts. So in theory, they can go in either direction. But in biology, right, there's usually a favored direction because of how the physiology is going. So now we can do this comparison and we can say, right, so based on what I think I understand about biochemistry, would I believe that the metabolite QTL and the pQTL should be in the same direction, that those betas should be in the same direction or opposite direction, so positive correlation or negative correlation. And then Josh's work from the colocalization, are they going in the same direction, opposite direction? You can see in the vast majority of cases, the directionality works out, which again is something I think special that we can do with the metabolite QTLs, where again, because we understand the biochemistry, we can say that that what we think is going to happen based on having more of that enzyme. Again, I guess I haven't talked a lot about how this is relevant to my work in drug discovery, but obviously, if you tell me, oh, target X is what you should go after for disease A, right? Well, one question, of course, is going to be what organ and what time of development and the rest. But the other question is going to be, do we need an activator or inhibitor of this, which is obviously going to be a huge thing. Often from GWAS, we don't quite get that information. This is showing that the pQTLs probably are giving us good information about the relevant causal gene and the direction of effect. And also, I find it reassuring that what we are measuring in circulation from both proteins and metabolites probably has some relevance to what's going on in biology. And that's the end of this section. I'm going to jump into a new section, unless there are questions about this. No? Okay, good. Whoops, going backward. Sorry. Let's go the other way. There we go. One thing which I think is not, well, actually, I'll be happy to see if this gets more discussion as we go. This is now one of my new current things that I'm trying to get people to think about, which is that especially, well, I see it especially in the area of metabolite QTLs, but I'm going to show you examples how I think this persists throughout. And Maria, maybe you've seen that. You must have seen this after going through 150 gene lists, that the same genes often will come up on multiple lists.
Hey, Eric, for sure.
Yeah.
And Eric, before you go farther, we do have a question that I think pertains to your last section.
Oh, good.
For directionality, did you subset to co-localizing pairs?
Oh, yes, absolutely. Yeah, this is only things that co-localize. Yeah. I mean, it's an interesting question whether we have any pQTL, metabolite QTLs, where I believe it's the same gene, but for whatever reason they didn't co-localize. I mean, those are interesting also because you can have different causal signals that both act through the same gene, but maybe under different conditions, different tissues or whatever. For this analysis, these were only those co-localized pairs, and only the co-localized pairs where I thought I understood the biochemistry enough that I could make a prediction. Yeah.
Thanks.
Okay. New theme, new topic, where after the subtitle of this is sort of lessons after looking at or annotating 100,000 GWAS loci. So certainly, and a lot of those, many of those, maybe most of those are metabolite QTLs. So this is an example from again from the MetSim work. But every time I look at metabolite QTLs, I see something similar. So this is the PheWAS plot, and there's the link if you want to go play with this now. So here the gene is, the gene I've assigned is GLS2 glutaminase. It breaks down glutamine. The strongest signal there you see is glutamine, but you also see all these other metabolites which have glutamine in their name, right? And it's probably not the case, or I guess I'm hypothesizing that it's not the case that the enzyme works directly on those other metabolites. It's that those are downstream effects of manipulating, affecting the glutamine levels. And this web up here is how I kind of think about it. If we think of glutaminase GLS2 here at the middle of its biological web, it has effects that kind of ripple out away from it. The most proximal effect to that is what it does to glutamine, and then you have these secondary effects around all these other metabolites. And so how does this play out? In that Yin et al. Paper, I didn't make this explicit, and I guess what I'm trying to do now here is make this a bit more explicit, is that I think an important part of interpreting a GWAS locus is to also ask what are all the other effects of that same variant. So we do talk about this metabolite, so N-acetylkynurenine, I think it's our figure 1 or 2 in that paper. Just a random, well, it's a random but nice, a nice case study metabolite. There's the structure of N-acetylkynurenine. This part up here is the N-acetyl group. This part down here is the kynurenine group. We have these 5 strong associations with it. This is the Manhattan plot. Again, you can go to the Phi website and look at this in more detail. To interpret these, if you take each one of these variants and then you do the PheWAS of those, what you find is that even though all these variants affect the same metabolite, the PheWASs for them look almost completely different. So each one of these variants is coming at the same metabolite, but from a different direction. And the information about that full palette of what each variant is doing really helps us nail down what the causal gene is at each of those. And so I'm going to walk through this just briefly. At this variant on chromosome 2, you see all these other metabolites that are N-acetyl this, N-acetyl that, blah, 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 blah. So all of them are N-acetyl amino acids. The enzyme that's nearby, this is actually the second closest, is this N-acetyl transferase 8, NAT8. Right, and so we would say, well, that's probably the causal gene because what it's doing is adding an N-acetyl group. The next one here on chromosome 11, and actually the other thing I'm highlighting is that N-acetylkynurenine at none of these loci is the strongest signal. It's maybe the 5th strongest or weaker. But what we learn a lot of additional information by looking at all the other variants that are at that same locus. And well, actually, maybe I'll leave it there because I'm realizing I have a lot more I want to cover. But we have this other one, this ACY3, these two that seem to be somewhere, well, if you look at the metabolites, they're all within tryptophan metabolism. And we have these 2 variants that are acting on, or probably acting through the kynurenine region. And then a locus that actually has a number of different amino acid transporters or organic anion transporters. So you learn a lot by looking at all the other associations at that locus. I'm going to skip over my HAL story. I love the histidine ammonia lyase story, but I'm going to skip over because I've talked about it a lot before to go to my new favorite, which is TRIM58. Again, I didn't get a chance to thank the folks at GWAS Catalog for hosting me over the summer. I had a great time out there. I apologize because I think I did show this story there. I tried to throw in enough new stuff to keep you guys interested. This is one that I debuted there over the summer. I really like this locus, this story. So TRIM58 has an association with HbA1c. Of course, that's a phenotype we know and love from diabetes. It's your glycated hemoglobin. Nicole Soranzo and others have shown that generally, right, HbA1c is a combination of hemoglobin and glucose. And so either modifying glucose metabolism or hemoglobin metabolism could be a way to affect HbA1c. So it's the TRIM58 locus, but what actually is the causal gene there? So if you go to the GWAS catalog and say, okay, what are all the other associations that are near to that glycated hemoglobin? So here it is down here. It's a very weak association. Well, okay, not very weak. In 2024, it's very weak because it's only 10 to the minus 23, right? But then there's a lot of other associations, including 10 to the minus 600, right? So you have all these other associations, and let's just take a quick look at these. We have a protein QTL, dynein levels. Then we have reticulocytes. Those are premature red blood cells. We have red blood cell traits. We have hemoglobin traits, and so on. So is there a through line that allows us to make sense of all of these traits? And in fact, there is. So there's a nice paper, and I put the citation down there. TRIM58 is a ubiquitin ligase. One of the things that it ubiquitinates, and this paper and among others show that one of the things it ubiquitinates is dynein. One of dynein's jobs is in every cell to help keep the nucleus in the middle of the cell. It's doing a tug of war with kinesin. TRIM58 in these erythroblasts, early red blood cells, its job is to ensure that dynein loses that tug of war so that kinesin can pull the nucleus out of the cell, resulting in a reticulocyte, which then becomes a red blood cell, which then produces hemoglobin, which then can accumulate glucose. And so what I'm showing here in these ovals here, each one of these is a GWAS association. In fact, the p-value, and we can't read too much into this, but the p-value of each of those associations gets weaker as we go further away. And so again, this is the sort of web story where TRIM58 is at the center of its web, and then it's radiating out from there. And so I don't see, and so again, my premise here is that in understanding this really weak association down here with hemoglobin, it becomes much clearer and a much better story if you think about what are all the other associations on top of that. And I think, again, I know some of the folks here are working on this already. I think it would be amazing to have this co-localization of everything against everything. So we're not trying to annotate individual variants, but we're trying to annotate the full PheWAS of each of these variants. This PheWAS, this variant, is having this total physiologic effect, and here's how it manifests across all these different traits we can look at. Yes? Okay. Good. Okay. I have two more topics, but maybe I'll skip the last one because I need to talk about this. I don't think Brent didn't make it. Brent's not there yet. Yes?
Yeah, not quite yet. His
Not
flight's
quite
a little
yet.
delayed,
Okay.
but he'll be here eventually.
Good. Okay. So this is work that I started with Brent and Anuba and Mark many years ago. The graphic here is picked obviously and intentionally because I think we made some nice progress for a variety of reasons. It's kind of been locked in place, and I appreciate this workshop to maybe help move this a bit forward. But I'm going to walk through how we sort of addressed and looked at this problem. In this case, I'm going to the specific gene I'm going to talk about. Yeah, this is stuff that I tweeted out once upon a time when Twitter was Twitter. My who's that causal gene. And in this case, the trait is hemoglobin concentration. You can see this is pre-Dali. I had my son draw what he thought a picture of hemoglobin concentrating might be. And that's what that is.
Pretty good.
But yes. So here, this is the locus zoom plot on that. There's a number of genes in the region. To give away the punchline, in this case, the causal gene is this EGLN1. It's a really cool bit of physiology where as long as you have oxygen in the cell, EGLN1 gets broken down. But as soon as as soon as an individual cell lacks oxygen, EGLN1 then persists and causes production, allows HIF1 alpha to persist and launch all these downstream effects to increase hemoglobin and other things. And again, going back to the fox and the nest, through this work, and coming back to your question, Noel, through this work, we sort of settled on just 4 fundamental types of information, or at least 4 that can be, and I will show you 4 that are relatively straightforward to accumulate for every gene trait pair. So mouse knockout information, rare disease information, drug target information, and this comes straight from open targets, and genes with sequence altering variants that also associate with that disease. I'm not going to spend a lot of time on this paper. Hopefully most people are familiar with this from the Regeneron folks. I mean, this really nicely showed that if you take all the rare coding variants and you just tag all those genes are far more likely to be in the vicinity of a GWAS signal, a common variant signal for that same trait, which again, I think nicely justifies using rare coding variants as a rationale for justifying a particular gene. And so EGLN1 is a nice example because you can actually check off each of these boxes. So yes, we completely understand the biology, and that's why I like coming back to it. I'm trying to understand what the biology is of each of these targets. But in addition to that, if you knew nothing about the biology and you just said, you looked it up and you said, there's a mouse phenotype with a relevant, that's relevant to this trait. There's a rare disease that's relevant to this trait. There's a coding variant in that gene that has that same trait. And in fact, there is a drug that targets EGLN1 that's relevant to this trait. And so what we did for this, and I'll walk through this in a bit, is all this now, and again, thanks to the work from open targets and EFO and so forth, this can be condensed quite nicely, where what this sentence, what this block of table is saying is that EGLN1 can be referenced or used as a causal gene for any one of these 4 traits. And again, because we have EFO, I don't have to spell out the full traits. I can just say, here are the EFO terms, and this is what they expand to. These are the labels for those. This is a sentence now that I have written that reminds me and others of why biologically we might believe that that gene could be causal for that. And then just a paper linking back to sort of the experimental work that would link that gene biologically to that trait. So all this information that I just showed on the previous slides of why this gene might be relevant to that trait is encoded here. Linking in now those other lines of information, and I think this we will come back to, and I've had many conversations with Jason around this, of how do you pick the right mouse phenotype? And I know other people in the room have also thought about this. Whenever I come back to this, the sentence is foremost in my head. I come back to this all the time. The criterion of biological salience is surprisingly inclusive. This is a great paper. They show that only one schizophrenia candidate gene actually reproduced in the GWAS, even though for every one of those papers, the authors said, this gene must be related to schizophrenia because X, Y, and Z. What they show in the paper is that those criteria are things like it's expressed in a particular region of the brain at some stage in development. That can apply to 2/3 of all genes, right? So you can justify 2/3 of all genes through that. So this is something that I do fret about when I look at papers when people say, oh, this gene must be related to that. I did have a whole tweet thing. So the NAT8 locus that I talked about, there's a whole paper saying, oh, EQTLs say it must be ALMS1, and ALMS1 has metabolic effects. I mean, again, half of all genes have metabolic effects. The PheWAS we see at that locus is N-acetyl A, B, C, and D. The only gene there it can be is NAT8. So I think currently in the literature, there is not good, there's not good standards on how specific the biology has to be to rationalize a gene to a protein. And I think we can get a lot better at that. What I'm showing here is if you want to rationalize EGLN1, you could say, oh, the mouse knockout has a homeostasis phenotype. Well, that's right, 1/4 of all genes. And in fact, right, if you look at every locus in the genome, 80% of loci have a gene from this category within the closest 5, right? And so if this is your criterion, that's not very convincing. These, of course, the MGI is a nested hierarchy. It's an ontology, so you can go finer and finer detail. down here you can say abnormal nucleated erythrocyte cell number. There's only 50 genes that have that, so you only have a 1% chance of finding one of these genes throwing a random dart at the genome. What I've done in this work is, I don't have a good heuristic for what level of specificity that should be. Just empirically, somewhere in the vicinity of 100. So if your biological criteria can apply to about 100 genes and no more, then that's probably pretty useful. And again, I know Jason's been playing with that, or you can play with that through the Pigeon tool. What I did do though, and what I would like to see is, I don't know if Helen is on. I credit her, Helen Parkinson, for this all the time. Is early on, she said, just be explicit in what you're doing. And so what I've got here is I can explicitly say, okay, I'm going to use these specific mouse traits to rationalize these specific GWAS traits. And maybe they're right, maybe they're wrong, but they're explicit, and here they are. And then we can look at what those numbers look like.
So
using
Eric, I have a
yes.
question. So this is getting at really like some great things we want to come back to. Like this is like how we create these lists, how we interpret them, how we use human inference and intuition to infuse these lists, if you will. I'm also curious if we have a mouse biologist in the room. Yeah, how you want to how like that community could actually feed into making these lists better because of what Eric was just talking about, like making the right choice about the mouse phenotype and whatnot.
Well, I think, Eric, you touched on it. The IMPC has done a beautiful job knocking out some number of thousands of genes. The phenotyping that they apply to those mice is a little problematic in terms of phenotyping 20 knockouts and expressing that relative to thousands of controls over the course of years. So really harvesting the full potential of that knockout data is a little tricky. I'm working with Jason and Noël on another aspect of this, where we've done deep sequencing on 5 key metabolic tissues in 8 different mouse strains that together contain roughly 40 million SNPs, which is comparable to the human population in genetic variants. And what's really amazing there is almost every, and it's EQTL, but we're working with Steve Gigi to do the proteomics, and what you've done for me here is sell me on the PQTL over EQTL. But I can tell you at least almost every single gene in the mouse genome is subject to genetic regulation, which is pretty amazing, right? And then working with Jason, and he may talk about this tomorrow, I'm not certain. If we take the human homolog of those genes subject to genetic regulation in those 8 mouse strains, a huge fraction of them are decorated with various phenotypes in human GWAS. So we're kind of doing the same thing, that we're exploiting genetic variants in the mouse to identify which genes are subject to differential regulation at the RNA level. Can we pinpoint that to metabolic traits that we've measured in those mice? And can we link it to associations in human GWAS? And it's an ongoing effort, and I hope Jason talks about it tomorrow.
Maybe we have a whole breakout on EQTLs, just in a quick nutshell. My interpretation there. So yes, I know EQTLs have high overlap with GWAS traits. My interpretation, and there have been several papers, and there was just one from the EQTL catalog folks just recently.
Yeah.
Right, there's high pleiotropy, right? So you have an enhancer, and it's affecting 10 different genes. And right, so you have changes in transcription level of those 10 different genes, but 9 of them aren't important, right? And only 1 is actually driving the trait. So yes, you might see this overlap of EQTL of gene A with the trait, but it's just because it's borrowing the same enhancer of the gene that's actually important. That's my interpretation.
And your instincts are absolutely right. These enhancers can be a little bit promiscuous.
Yes.
And it might be like what's bound there. In terms of, because these are not single transcription factors that sit here, it's a whole conglomeration of them. And we've discovered that, what might be there or not is a function of the environment. That is, what diet is the mouse on? Which is not so surprising that diet could regulate the level of a particular transcription factor that may or may not sit at an enhancer and that determine what effector gene it has. So you're absolutely right. and I love hearing the story about the PQTL.
Yes.
Co-mapping with metabolites, that makes sense.
I mean, I showed my, right, I started in my science career with protein structures, and then I went through this whole human genetics piece, and I'm so thrilled now that we're coming back to proteins. Gives me a lot of pleasure. I guess I won't have time to do justice to this slide, except to say what I have captured now are these bits in blue. So things like saying, EGLN1, right? So I'm saying that I'm stipulating that EGLN1 is a causal gene for these traits. Here's my human sentence, human readable sentence for why that is. Here's my paper justifying that. All these other bits now are added sort of automatically because of these other heuristics that I've applied. And the heuristics are down below. Again, I'm not going to be able to go into detail, but to say that what I'm saying here is all these EFO human GWAS traits can be rationalized by any of these mouse traits. And then the rest is just added, right? So this is just looking up what those EFO terms map to. This is looking up what those MGI traits map to in terms of text. And this is now an automatically generated sentence based on that, which again is just what I said out loud. A claim that a gene is causal for the GWAS trait of erythrocyte count, blah, 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 can be supported by evidence that the mouse knockout of the ortholog of that gene results in blah, blah, blah. Yeah, and similar for rare disease or drug information. So at this point,
Here, we're about 5 minutes, but we'll give it.
Yep. Yeah. So I'm going to skip my last topic. So I'm going to finish on this. No, I've got it. I've got my clock right in front of me. So I've done the metabolite work here and the protein QTL work. The 78 diabetes, that's the piece coming from Mark and Anuba. The bone mineral density, that's work with Brent, and then others on the team to help some of these. And this now comes to your question exactly. So applying these different sort of automatic rules and heuristics, you can see for my metabolites, I can automatically accumulate coding evidence for 272 of them, mouse knockout evidence for 200 of them, rare disease evidence for 164. You can see it changes by category. I haven't found any, the pigmentation loci I love because that's sort of where mammalian genetics got started. And so those are very well known and they all come up in human genetics. I haven't found a drug that acts through any of those. But this is kind of the table of these 1,700 rules. Again, each one of these rules says this gene, if you find it in the vicinity of that trait, right, if you find a SNP for that trait in the vicinity of this gene, then you're justified in calling that the causal gene. This is something that we'll also come back to. The original list that I provided or generated were always a SNP, a gene, a trait. I've moved away from that because I think that it's more generalizable, right? So EGLN1, if somebody does a study in a new population and you find a trait, you find a red blood cell trait for a variant that's never been seen before, but it's near EGLN1, that's probably still the causal gene, right? You don't need to know exactly how it's linked. What you need to know is that EGLN1 has that sort of effect. What I'm showing here is if you go through the entire GWAS catalog, these are all the associations from all the different studies that can be linked to EGLN1. The way I say in the vicinity, currently I'm defining in the vicinity to be within 500 kb, but that obviously can be changed on the fly, which is another advantage here. But what I'm showing here actually is that the strongest associations for which EGLN1 is designated the causal gene. So this is distance to EGLN1. 0 means it's within the EGLN1 itself. And here are the traits. This is the p-value of the trait. The strongest traits, the strongest associations are the ones that are actually closest to the gene. And we can talk about why that might be. I just have to, in my title, I said, or actually it's still up here somewhere, 100,000 loci. If you apply these 1,700 rules to the 500,000 associations, lead SNPs, and GWAS catalog, about 100,000 of these can now be assigned if you believe the sort of the rules as I've laid them out. This is what it looks like. So, and this is a complicated table that I won't have time to give justice to, but there are 463,000 as of end of June associations with a p-value of at least 5 × 10⁻⁸, where there is a chromosome and a position listed in the GWAS catalog in those fields, and 100,000 of those, I think I know what the causal gene is. That's about 20%. Up in the other corner, I'm just highlighting that of the metabolite QTLs with a p-value of at least 10⁻⁴, I think I've got the causal gene for 90% of those. The curve, if you take the strongest association for each gene, and at this point there are 676 genes for which I think I know at least one GWAS trait. My goal is a GWAS trait for each of the 20,000 genes. So I'm working my way up there, but this curve should look familiar at this point. I'm going to skip. I'll show you this picture just because I love this picture. I'm going to skip this whole thing about using AI, but to say that I think, yeah, the gene closest to the lead SNP usually is the one that makes the most biological sense. I'm going to skip this story, even though it's a fun story. But come back to, here we go. I'm going to close on this, 1 minute to spare. Hopefully I've convinced you it's usually the closest gene, or at least you need to compare your method to closest gene. In a lot of the work that I've done, the mouse phenotypes are great. I have not had much luck in finding other model species that have data organized in a way that you could use it the same way, but I would imagine rat, zebrafish, others should be useful, but I just have not found a good way to mine that data. You know, there are 500,000 associations in the GWAS catalog, but as we'll hear about, I mean, the same genes come up over and over again, and so you don't need a huge list. I've got now 1,700 of these rules that can explain about 20% of all of those. And this last point here, I do hope that we move more towards thinking about annotating the PheWAS. And so, you know, for each of the diabetes traits, I mean, this maps to a lot of the work that we've seen from a lot of folks, that it's not just a diabetes locus, it's a diabetes locus that also has effects on lipids and so forth, or it has effects on insulin or something like that. So I will stop there.
Awesome.
And thank you, everybody.
Thank you, Eric. That was fantastic. We'll have virtual clapping. So I have a couple questions. I know Laura does. So Laura, do you want to start?
Sure. I was thinking about you were talking about the standards for biological salience, and that there wasn't a good heuristic for that. And this is not my area of expertise at all, but I was just thinking, you know, when I was a postdoc a long time ago and I was doing gene expression work, and there were statistical methods for kind of assigning pathways to, you know, the variants on your array were overrepresented, and that were significant, were overrepresented in certain pathways. And I'm just wondering if there are statistical methods for how reliable that evidence is, and, you know, maybe that's it, that other people on the call might have thought about this too.
Yeah, like I say, and I guess we're out of time. Otherwise, I was going to put Jason on the spot if he's in the room. I mean, this is the exact conversation I've been having with Jason. I really love his Bayesian approach. And is there some way we can leverage that? Now, I think I've asked him 4 times, and he's explained to me why it can't actually be applied that way. But I'm still hoping to find something there. Again, I'm a big fan of the pigeon approach. And I mean, clearly there's different levels. You can assign different levels of information value, right? If something applies to all genes, it must have less information value than something that is very specific. But is Jason in the room?
He's not yet. He's en route. So we'll
Okay.
save that question for later.
Yeah,
Oh, there he is. Yeah.
just really quickly, I think that if there's a short answer, is an annotation that affects on the order of around 250 genes seems to be kind of the optimal size to your question, Eric. And but the way that we have worked on estimating this is looking at how enriched given gene annotations are for GWAS hits. But it's not possible, as Eric mentioned too, it's not possible to convert that into an optimal size without some prior assumptions on that. So again, not have time to go into it fully, but I think your intuition is correct, Eric, that you don't want something that's too pervasive. Typically around some annotation that affects around 250 genes or so seems to be around optimal.
Sounds good.
Okay,
So
thanks. Do you want to go ahead now?
Yeah, I have one more question, Eric. So like what you, I think it was slide, I don't know, 3 slides ago, we had that big table for the 1,700 genes to trait. That in itself is an effector gene list in my mind. So I just, back to our theme, if you were to make this accessible and a resource, you need some standards to hold yourself to. I think it's, go back, no, it's a bigger table than that. Which one was it?
Well,
Go back one more, maybe?
no, so this one?
Yeah.
Yeah.
That one. So, you
Okay.
gonna say something? Yeah.
Yeah, well, so I did, right, so this stuff hasn't been published, and yeah, I hope to chat with Mark and Brent about, yeah, how to push this forward. I did take a baby step with this, and Maria helped with, so the EGEA list. So that's my internal label for ever annotated, right?
Yeah.
So what I put out in GitHub, again, I'm within a company and it hasn't been published. So what I did was I took the subset of these 1,700 that have been published and I formatted them in basically the same way. So what's currently on the portal under, I forget what it's labeled as, Eric or Eric's list, is basically the subset of
metabolites, yeah.
Yeah,
I think
it's the
based on
subset of
phenotypes,
these,
metabolites, yeah.
yeah, it's the subset of these 1,700 that that have been published, basically.
Cool.
That's only though, so I think there are three, well, no, there's two important lists. So one is those effector gene lists. The other, and I don't know if this is on anybody's radar or how we want to do it. The other is, sorry, this bottom part here. So the top part I call the gene trait assertions, right? So somebody has said, Eric has said, EGLN1 is a causal gene for X, right? So that I think fits into the effector gene list. But again, I hope we have some conversation about whether those effector gene lists must have SNPs with them or not. The way that I've gone is my initial list does not have the specific SNP. And then I go through GWAS catalog or whatever and say, here's all the SNPs, all the associations for which that applies. This bottom half here, I call valid evidence rules. And I have not seen that before. And hopefully other people are thinking about this. Again, this is, if this is the trait that you're trying to justify, here are the mouse knockouts, the rare diseases, or the drug indications that can be used to justify that gene. I have not seen that standardized at all. And
that's exactly what I'm kind of getting at, because I think that's really critical to make these more useful to the broader community, and is something that we could help as a community develop standards or at least a structure for, so they can be machine readable, they can be human curated, they can be used in other settings. So I think this is brilliant to share this.
And again, those first three columns, right? So I mean, I call it, well, the first three columns I call manual because I've compiled these. But obviously I'm going for machine readability because you can see each one of those, and the trait ID column comes from EFO, the valid evidence. Actually, most of this is somewhere in the ontology browser that EBI hosts. So I think all of this can be done through resources that EBI has at this point. And then those bits in beige on the right are the bits that are added programmatically. So right, just going through the blue bits and then generating those automatically.
One comment, and Butterworth will just read it out loud. It's kind of building on this topic of biological salience is clearly a highly predictive method for metabolite QTLs. But as you highlighted with the schizophrenia paper, biological salience is a considerably horrible predictor for complex disease GWAS signals. Does that imply that we need different rules and possibly different standards for different kinds of GWAS phenotypes? That's a great thing to put out there, Adam. I love that. Eric, do you want to take a stab at that question?
Sure,
Or
I'll take a stab at it. And maybe we'll come back to this at the end also.
Yep.
I mean, I've been playing, I've been talking to Jason, playing with the Pigeon tool. I think what we're seeing there is that for many traits, and maybe we come back to this, and maybe Jason, you have a better sense, and we can come back to this, about the types of traits that it works for well and it doesn't. I think for a lot of the traits, they do behave well. Height coming back to skeleton and so forth, and I mean pigmentation. Where I have not spent a lot of time is psychiatric traits. I think and again, that's what the paper is about. Schizophrenia. I've stayed very far away from educational attainment and most of the psychiatric traits because I think there just aren't a lot of good methods or rules there. So I've pretty much stayed away from those. I think right. I mean, you can see from the list. I mean, we do cover IBD and bone mineral density. And those do, I think, work pretty well. I think mouse knockouts, rare diseases, I think do apply there. I mean, certainly, I mean, you can see in the effector gene list, maybe we'll come back to this. You know, when people do annotate metabolic disease and a lot of the inflammatory diseases, rare disease lists and mouse knockouts come up there. I see that Jason has his hand raised.
All right, Jace, one question, then we're gonna move on.
Oh, just really quick on that. I would, at least looking at the annotations, certainly for the metabolic disorders that you're mentioning, Eric, like the mouse phenotypes work really, really well, and those work less well for the mental disorders. I would assume that a lot of the different standards might just have data availability. Like it might just be that the mouse phenotypes aren't as good for some of those mental disorders. So as I said, we definitely do see it for the annotations. And I would imagine it would be similar for some of the other predictors in which the cell type and tissues might matter.
All right. Thank you, Eric. That was really great. And I think that set us up for some good discussions and questions we'll do later. I want to move on to our next talk from our Open Targets partners. Yakov, are you there? And shall we start to have you share your screen?
Yes.
Yeah, Yakov's here in the room with us.
Yes,
Very
I'm here.
So I'm hoping
Hey.
the camera is going to zoom in on you, but if it doesn't, maybe we can make that happen.
I'm currently sharing my screen. Can
Yes.
you see it?
Yeah. Again, for Yakov, I would put questions in the chat. If there's something that is like burning, I will stop you, or you guys can stop. Feel free to unmute yourself if you want. You know, this is interactive. So with
Okay.
that though, Yakov, hit it.
Well, let's start then. First of
Yeah.
all, thank you very much for inviting us to this workshop. It seems very relevant to us because one of our main streams of work is basically predicting genes in GWASs. I'm anticipating quite a productive discussion, and I hope that we have a lot of stuff to add into it. So first of all, about Open Targets. So Open Targets is a private-public partnership between two academic partners, CBI and Sanger, and 5 industry partners, represented by 5 pharmas. That is focused on providing the comprehensive platform for target prioritization. Open Targets does a lot of stuff, but probably one of the main focus is provide the comprehensive genetic evidence about the targets to prioritize for the specific diseases. So the main focus is genetic evidence. There are a lot of types of the genetic evidence. Open Targets works with probably all of them. I'm going to focus on genetic evidence that goes from GWAS, and this is exactly the topic of workshop. And the unit in Open Targets that works with the genetic evidence from GWAS is called Open Target Genetics. So I'm going to focus on this. Anyway, this topic is going to be more general and focused on technical side of the question. More details about what we want, what we currently have is going to be presented by Jack from the team tomorrow. So this presentation is going to be only like about the concepts and definitions. This is an example of association page in Open Target. So if you have the specific disease and you want just to visualize like the all possible evidence for your target, this is how it looks like. The first 9 columns, or how many of them, is actually dedicated to germline genetic evidence. So you can see there are a lot of different sources. The GWAS one is represented by basically only one column, Open Target Genetics. If you click on it, it will just simply visualize you the information about the genetic evidence from the particular GWAS studies that are incorporated into this, if you have any. This all descriptive information and our prioritization score that is called locus to gene, or L2G in short. I will explain later what does it mean and how we actually calculate it. So how we actually work with the genetic, with GWAS data in our platform. This is probably the main slide in this presentation. It's kind of difficult one. We have 5 different edges of information about how we actually represent the GWAS information. So it all starts from basically the disease. Since we work with GWASs on systematic level, we are not focusing on any particular disease. We work with whatever we have. All GWASs are mapped into the ontology. We use if ontology that is kind of like whatever is common to use in API. So this represents the first edge of information. The second one, within each disease, each E4, we have at least one study that represents GWAS. Each GWAS is converted into credible set. I will stop here for a moment because this step is basically, well, probably we are on the same page with all our partners here, because I think everyone now applies kind of similar approaches. So how to basically represent and reduce the information that is stored in GWAS. Credible set is next step from the very old idea of representing GWAS just as a list of lead variants. For example, like 10 years ago, people just tried to represent GWAS as a list of lead variants and all their proxies. It works, but credible sets, given how advanced the genetic and fine mapping technologies are, it's basically just the next level. And it represents the table of the variants within the region with their corresponding posterior inclusion probabilities. These credible sets, they can be small, they can be large. If you are lucky enough, you can have credible set with the size of 1 that basically represents that you have the exact, probably, well, semi-exact answer that this exact variant is probably causal 1. Each credible set represents 1 causal variant. So if you have 10 credible sets from GWAS, it simply means that you expect to have 10 causal variants. So it's kind of a simple representation, but it slightly contradicts with whatever people used to present in all effector gene lists, because usually people just represent the lead variant and say like, okay, we have lead variant and we have this gene. This is a very simplified version because, again, credible set can be very large. What you can do with the credible set that has the size of like 1 megabase, of course, you have like, you can imagine how many genes you have in the region, and it's very simplistic version to just represent this credible set by 1 variant. But we will return to this question. Anyway, GWASs, credible sets. Fine mapping is the most expensive and difficult part of the pipeline. I'm not going to stop here. I'm not going to give any technical details on the procedure. We are trying to use all state-of-art approaches, ancestry-specific fine mapping and everything. Just assume that we have credible set. Clean one, proper one. Then for credible sets, we simply define the list of genes in the region. And for each gene, we are trying to get the list of functional genomic features. So like everything we can do, starting from very canonical ones like distance-based features, colocalization, this expression of genes in relevant tissues, variant effect prediction, intervals, whatever you want. Like we have many features, and all details are going to be given tomorrow in the presentation of Jack. Then when we have this table that is in this presentation is going to be called credible set level feature matrix, we can estimate the prioritization score. So basically for each gene, for each credible set, for each GWAS, we have like very specific number that spins from 0 to 1 and represents how likely this gene should be prioritized. This score is in our platform is called locus2gene score. So this slide just simply visualize how exactly we create the credible set level feature matrix. So in this very dummy example, we have like credible set with a size of 4. with a lead variant, and it's probably like going to be used as a sentinel variant for this credible set. We have a lot of genes, so we simply select all genes around all variants in credible sets. So again, if the credible set is big, if it has like 100 variants and it has like it spins for 1 megabase, you are going to have a lot of genes. And yes, obviously this credible set is not really that informative as, for example, credible set with a size of 1. But anyway, credible set, corresponding genes, and for each genes, you just simply populate the columns with the features you have in your model. In our current model, we have around 20 different features. So this is just an example, dummy example, how it's supposed to look like. But in the very end, after you apply the whole machinery GWASs, you are actually going to have quite a large matrix that will consist information about all credible sets in all GWASs for all genes. In average, you from our previous results, you have like 15 genes per credible set. Actually, now it's much more because we analyze all genes around the credible set, not only around the lead variant. So you can imagine that the feature matrix is pretty huge. At this moment, we actually can stop because it already provides quite a comprehensive information. Like this is a genetic evidence itself. You can show it to any human geneticist, like to make a manual decision. Like, okay, guys, I see this feature matrix. I see that this specific gene in this region and this GWAS is probably like has much more functional genomic features than other one. So I probably should prioritize it. But we try to, well, it happened 5 years ago, actually, not we, to apply the machine learning approach and to estimate the prioritization score for each gene separately. So how it's possible to do? Many ways how we can do it. I will present probably the easiest one to explain. So again, imagine we have this huge credible set level feature matrix. We have all EFAs, we have all relevant GWASs represented by studies. We have all credible sets within the study. So in this very particular example for one specific E4, we have 3 studies. And in these 3 studies, we have 6 credible sets. For these 6 credible sets, we have 5 genes. So here color represents gene that can be met in different credible sets. So if you want to train your model, you basically need to decide which study or which credible set to use. In the logic I want to present, we decided to get rid of situation when you need to decide which credible set of study to use and just to use everything. So we can apply
Couple questions. Sorry
Yes.
to interrupt you. I just want to catch you because they're like quick questions. What's the maximum distance do you allow? And then all genes or only protein coding genes?
That was answered in the
Oh, it was
chat.
answered, sorry. Okay, sorry. Catching up. We're all good.
Currently,
It was
we are
answered.
working,
It was answered.
currently we are working with all genes, not only protein coding ones, and the maximum distance we allow is more like not more than 500 kb from the variant, not from the
Awesome.
credible set. Yeah, but distance is calculated as a weighted sum across all variants because we are not calculating distance to the lead variant, we are calculating the distance to the credible set. And we have 2 types of distance, at least 2. One of them to transcription start site, the other one to the footprint of the gene. But again, it's kind of flexible thing. So it's not really something that is currently fixed. We can work only with protein coding ones. Anyway, the general idea of this exact procedure, if you have several genes within one IFO, just simply collapse of information about one gene together and always select the maximal value for this specific feature. or the best value, I would say. For example, if you have gene 1 3 times, and for 2 credible sets you have 0, and for 1 you have not 0, just use 0 because this is the best value for this gene. The same goes for other features you have, like if you have the evidence of colocalization, if you have evidence of variant effect prediction of having missense variant in this credible set or anything else. So this big table actually becomes much smaller because now each gene is only presented once. So we call this matrix as disease level feature matrix or EFO level feature matrix. And again, it's can be provided to the community as a final product of the genetic evidence production. But again, returning to our previous question, how we can apply the machine learning to this. So having this matrix, it's actually quite similar, quite simple. At this moment, if we have effector gene list, so whatever it means, for us it's basically just EFO gene pair where you have like established known association between a gene and this particular disease. You can join it with disease level feature matrix, and if you have this call like this EFO gene pair into your disease level feature matrix, it's going to be served as something that we call gold standard positive. So something that they are actually trying to predict. Then using the distance rule, we can select other genes in the region and consider them as not associated with the disease, and we will call them gold standard negatives. So for now, you have like numeric feature matrix. You have the vector of, or the binary vector of zeros and ones that represents whether it's gold standard positive or negative, and you can apply any machine learning approach you want. We use XGBoost, and we call this model L2G, locus to gene score. And this model can be applied to basically any gene, any credible set. So you can apply this model to credible set level feature matrix, and you can apply and you can obtain this prioritization score. So that's simple. That's how we work with the genetic evidence. Just as an example, we also visualize it. So as soon as you have like these results, you can visualize not only the genetic, like the feature matrix itself, like the genetic evidence you have, but also you can visualize the prioritization score for the just to give this final decision to the user, like whether you should prioritize it or not. Again, returning to the general visualization, these prioritization scores, they are aggregated on the level of the disease, and they are visualized as one score in the association page in Open Targets. So numbers. These are numbers from the latest release in June. Since the main unit of analysis in our pipeline is credible sets, so all results are presented in the light of numbers of credible set we have. For GWAS related credible sets, we have almost 600,000 credible sets. It's quite a lot. For example, if you will analyze only ClinGen related credible sets from 10th release, it has only 14,000 credible sets. So you can see we actually have quite a lot. The main source of GWAS as we use is from GWAS catalog. We use GWAS catalog curated studies, top hits, and we use GWAS catalog summary statistics. We apply fan mapping on them and obtaining credible sets. We combine this.
There's a couple quick questions, but we're also running for time. How much more do you have? How many more slides do you have, Yakov?
It's like probably I have the summary is the next slide. That's it.
Perfect. All right. Let's do that quick.
So we combined it with molecular QTLs, and we have more than 2 million related molecular QTLs, including PQTLs from UK Biobank PPP, from some single cells in UQTL catalog, and etc. And yes, summary. So open target genetics, first of all, it produces the genetic evidence as a feature matrix, as a credible set level feature matrix or disease level feature matrix. It estimates the prioritization score for each gene. and it visualizes the results of prioritization. We incorporate other sources of the genetic evidence as well. So if someone actually does this job for us, we are actually happy just to simply incorporate it into our platform as a separate column, for example. So for workshop related thing, we have probably two questions. And one of the most important, what is the effector gene list? Because in our case, effector gene list is basically established effector gene pair. We are not asking question about how credible it is. We actually already like assume that if this pair is presented in this list, it's kind of credible. Or maybe it can be like more general and can be represented as a feature matrix in our case. So you basically represent the genetic evidence for all genes in the region. Also possible. But in this case, if you present it in the region, you need to provide also information about the region or about credible set if your tool set is around credible set, because you can't represent credible set but just one variant. It doesn't work like this. How we can use it? First of all, we can directly, well, if there is in the future, if there is a database with effector gene list, we will simply incorporate it into open targets. It will be just like a column, very convenient for us. Or we can use it directly in our training model, so it can be used as a gold standard positive list, and we can enhance our prioritization scores. So that's it.
Excellent.
And acknowledgement slide.
That was beautiful. Thank you, Yakov. Virtual clapping as well. There's some great questions in the chat. We're going to capture those and ask those later. But Yakov, you can answer them directly after you're done, because I think they're directed at some of your actual slides. It'll be great. I think we're going to move on really fast right now just to make sure that we capture everyone who do leave some discussion time. So I think our next speaker is Maria. Is that right? Or is it break?
We have a break.
We have a break. Let's get a break.
Yeah.
That was a lot. To
Thank you so much, Noel. And I just want to thank everyone before I start. This workshop has been a long time in the planning, and I'm just so happy that there was so much interest in it and that we're having great discussions already. And so I'm just really happy to be here. So before we talk about where we should go from here, I think we should talk about where we are today. And I've been doing some work over the past couple of years, I think, to try to figure that out. So let's get right into it. So it's not advancing far.
It's a little slow at first, but then it
Yeah.
goes, yeah.
Okay. So I just want to talk about a little terminology before I start, so we're all on the same page. So in our group, when we talk about predicting effector genes, we're talking about it as a post-GWAS activity. So people will do a GWAS, they'll find the significant loci, apply all these methods and data types, and predict which are the causal genes at those loci. And so a little more terminology, we talk about PEGs because it's easy to say, predicted effector genes. And so here's just a stylized GWAS locus with the associations and some genes in the locus. And so in early GWAS, people would usually just say the closest gene is probably the causal gene. And as we just heard from Eric, that's a really great assumption. But it's not always true. And so people have begun to do more gene prioritization. And when we say gene prioritization, we mean this activity where you take different types of evidence and you find out how much evidence there is for all the genes in each GWAS locus. And then sometimes, but not always, which is why we have this dashed arrow, people summarize gene prioritizations in what we're calling PEG lists. So this would be just one summarization of all the evidence for all the genes, and there's often a scoring system to say which is the most likely to be the causal gene. So a little bit of history, we got into this area back in 2019 when Anuba Mahajan and Martin McCarthy made a list of PEGs for type 2 diabetes. And it was a major goal of our consortium, the Accelerating Medicines Partnership in Type 2 Diabetes, to implement this list in the knowledge portals. And those of you who've been looking at the portals for a while, this will be a blast from the past because this is the way it looked in 2019.
That's right.
And if we're doing personal notes, I can just say that it was my husband, Ben Alexander, who made this visualization. But so Anuba and Mark looked at 3 different categories of evidence for predicting effector genes: genetic, regulatory, and perturbational. And then within each of these categories, they looked at different types of detailed evidence. So for example, genetic evidence has 6 different types of genetic evidence that were considered. And they also had a detailed heuristic for combining all of the scores for genes in all these different categories and coming up with an overall categorization of the likelihood that the gene was causal or strong or moderate and so forth. So it was implementing this list that got us thinking about how useful these lists are for the community, and we decided to start looking for them and curating them and representing them in the knowledge portals. And I'll talk more tomorrow about how we do that in the portals. But it also got us wondering, is this activity actually increasing? And what do people do when they make peg lists? So I started looking a little more systematically. So since we're defining this as a post-GWAS activity, I wanted to start with GWAS papers. So I took papers that GWAS catalog had loaded from a 10-year period. And I scanned the titles and abstracts manually. I am a biocurator, and this is what I do. And I looked for mentions of gene prioritization. And people express this in different ways. They talk about finding causal genes for a disease. They talk about establishing the genetic architecture of a disease and so forth. I cast a wide net. And then for those papers where there was a mention, I scanned the full text. And I ended up with 169 papers in which systematic gene prioritization had been performed. And this covered 157 traits. And so our criteria for systematic gene prioritization, the major criterion was that they looked at all GWAS significant loci. I mean, for almost every GWAS, people will pick out a few genes of interest and go ahead and look at them, often by going all the way to wet lab functional characterization. But our criterion here was just that they looked systematically across all the GWAS loci, and also that they looked at multiple evidence types. So we wanted to know more about these papers. So first of all, is there the number of these studies increasing? And I think we can say yes. So here's a graph over time. The blue bars are the absolute number of the papers that I found, the gene prioritization papers, and then the red bars are their percentage as a percentage of the total papers loaded by the GWAS catalog, which of course is also increasing every year. So it looks like there is a trend to do more of these studies. Okay, now I want to talk a bit about evidence. Even though in this workshop as a whole, I think we want to focus on standards more than, we don't want to tell researchers which evidence is best. I mean, you'll all have to fight that out for yourself, and it's an active area of investigation. But looking at these papers, I wanted to get an overview of what kinds of evidence have people used. So I categorized the evidence. This is kind of similar to the categorizations that Eric talked about a little while ago. So one major type of evidence is variant-centric. So in this approach, you start by identifying the causal variant, and then you look for evidence about how it impacts the genes around it. And so the nearest gene would be one type of this evidence. But some other types are chromatin conformation. So does the causal variant contact the promoter of a gene? Does the causal variant lie within a region that's annotated, say it's a promoter or enhancer for a gene? Also QTLs, of course, does the causal variant impact molecular properties of the gene, such as levels of its transcript or its protein product or its distribution of its splice forms? And for all of these evidence types, the last three evidence types, if these occur specifically in tissues that are relevant to the disease in question, that strengthens the evidence. So the other major category of evidence that we considered was gene-centric. So in this approach, you start just with the genes that are in the GWAS loci and look for evidence about their function. One major category for this is built by association, which I love this phrase. I think it's really useful. I didn't come up with it, but just the idea that a gene that's involved in disease is going to have relationships with other genes that are involved in the same disease. And maybe they're co-regulated, or they belong to the same gene set, such as pathway, or the proteins interact, or there's differential expression in disease tissue relative to healthy tissue. Another type of this evidence is perturbational. So if you mutate a model organism ortholog of a disease, or the human gene in a cell line, for example, do you get a phenotype that has some relationship to that disease? And also, are there any Mendelian disease mutations in humans in the gene that cause similar phenotypes to the disease? Another category is gene burden. So this is just looking at the aggregate associations of all the variants in the gene. And you can look at common variant or rare variant associations. And the idea is is, does the gene level score show a significant association with disease risk? And finally, there's literature and online resources, which are a very valuable source of course, really detailed experimental information about just specific genes and proteins. And then another category of evidence that I looked at in these papers was pipelines. And here's just a list of some of the most common ones I found. And these pipelines themselves take in multiple types of evidence as input. But they're very often used as in concert with the other evidence types, and people will run a pipeline, but then also aggregate other evidence. So few questions about how people do this. So how many evidence types are used per study? Well, that's a nice distribution. Most people use about 4 of these different evidence categories. I looked at trends in use of the different categories, and here QTLs were the big winner. Most of the gene prioritizations used QTLs, and the nearest gene was a close second, but it wasn't universal. I think Eric might have something to say about that. And I was a little bit surprised that perturbational evidence is not quite so used, because it seems to me very valuable.
Maria, did you separate EQTL from PQTL for that?
I did not in this.
Okay.
I do have the granular data, but for this overview, I didn't separate the two of them, yeah. But I can say that EQTLs are much more often used than PQTLs in the list that I looked at.
Sorry, P or?
E is more often used than P.
And then where are it like model systems? Are they lumped into perturbation?
That would be perturbation.
Okay.
Okay, and then another characteristic of these papers was whether they did, they all did gene prioritizations, but then did they put them in a peg list? And before I go ahead now, I want to stop and say that I'm going to show figures and files from real papers, but I'm not going to say which papers they're from, because my intent is not to call out anyone and to say, oh, you could have formatted this better. That's the work that's been done in this field so far is amazing, and it's a wonderful foundation. And I think we can only, by making better standards, we can build on this. So, so all the papers did gene prioritizations, but kind of surprisingly to me, 25% of them did not integrate those the results into a peg list. So on the left here, we're showing there's one study where they looked at 6 different evidence types and ranked the genes for each of the evidence types, and they're all in a separate table. So it's not easy to see, to get an overall conclusion from that. 75% of papers, however, did integrate all the evidence into a peg list. And this is an example of a beautifully organized and color-coded one that was really, really easy to work with.
Maria, here's a silly question. So for the papers where they did not incorporate their various lists, how did they summarize their findings? Like, was it just text or description, or did they just pick was it typical, like, were these papers more about the actual evidence sources or GWAS, or they're like about a specific gene? Like,
I would say in these papers, typically where they didn't summarize everything, they did this analysis and then they picked out a few genes to go ahead and study in much more detail.
so it's almost like they did this more as like supplementary to make their decisions about what they would pursue.
Yeah, I would say that it's like their objective was not really to make an overall peg list.
Right, I think that's an important point, because a lot of times, that's why these lists are sort of overlooked as an integrated resource, because it's more that the people that are generating them have a specific goal in mind. Like, we want to pick 5 genes we want to functionally characterize, right? So I think that's an important point, because I think that sort of feeds to 2 things. One is like, how do we make it easier for folks that are doing that, who are using this as like their secondary outcome, to share those results with a resource? What standards can we help them with so that it's easier for them to do that, so that they could make it into interactive? You can even imagine, I don't know, I'm just thinking this, if we were to allow people to ingest the different separate lists, we royally across GWAS catalog and the knowledge portal could actually integrate them and produce lists for them, right? So this is some of the things we should be thinking about. And I think we as resource builders think about these things, but you as consumers and contributors think about how best we would do that. So sorry for that aside.
No, thanks, that's good. Okay, so again, looking at these peg lists, some of them are presented only as images. And so on the left here, we have actually a table in image format, which as a biocurator just really pains me, because I don't know of any way to extract the information from this image other than retyping the table. Maybe there are some.
Can ChatGPT do this?
I would not trust ChatGPT to do this.
Actually, there are some OCR work that's being done to extract information from figures and.
What's OCR stand for?
Oh, sorry,
Optical
optical
Character
character
recognition.
recognition. So
Got it.
there's like a resource of doing that from pathway figures.
Oh,
Comparing
cool.
those to gene sets. So I'm assuming they actually probably pulled
Okay.
out the genes from that table.
There you go, something to think about.
Yeah, but
And
that's kind of the exception that proves the rule, right?
okay,
And
so
then
you-
apparently
so they
Laura
use
says,
that.
yeah, you're a PMC extracts info from tables as XML.
Cool, okay, well, so maybe I didn't have to give up on that table.
Oh, no, you sound
Tables, what is that? I think she's referring to tables.
Yeah,
This is an image of a table.
Yeah,
which
this is, yeah.
even
this is-
harder.
Yeah,
Yes.
all right. And so some papers present beautiful graphics, which I really like. I mean, they have a lot of information. For example, this one here in the middle, the colors mean something, the angle of the little symbols mean something. And then on the right, there's a nice circular diagram of different kinds of evidence. So I think graphics are great. I just think they should be presented with their underlying data.
So that's one thing we should think about later, what other types of graphics that we can represent, so that's a great point. Like that, keep going, sorry.
So another major difference within these papers, with the PegList papers, was that some of them present all genes in a locus, evidence for all genes in a locus. And here's an example. This is every row in this spreadsheet I'm showing is just a gene in one locus, and evidence types are color coded for the strength of evidence. And so 71% of papers did this, but then the rest of them only presented the top gene per locus. So here's an example of another really nicely organized peg list, but it only gives the top gene in the locus. You don't really, you're kind of losing the evidence for the other genes in that locus.
We're all sort of cringing, hoping that if you show one of our
It's not gonna
figures,
be yours.
it's a good example versus a bad example.
Oh dear, no, there's no bad here. Okay, another big difference was whether there was a scoring system. And so this is the one I just showed, but I showed it again because it does have a scoring system. So, but again, somewhat surprisingly, only 29% of papers used a scoring system of some kind. It was most often just adding up the lines of evidence, but sometimes there were more complicated systems for combining the evidence. Here's an example where there was no scoring system. This was 71% of papers. So here, this is just one row from the table, and this is for one locus. So they looked at all these different types of evidence on the top, and they just listed which of the genes in the locus had significant amounts of that kind of evidence. So again, it's pretty hard to get an overview of which is the most, the gene with the most evidence in a table like this. So finally, I wanted to do some comparing of lists for the same trait, and there actually aren't too many lists where that are comparable for the same trait, because a lot of the lists that are where there are multiple lists for the same trait, they often come from the same group that keeps on refining their GWAS and refining their peg list, and so comparing those is not really that informative. I wanted to find lists from different groups that we could compare. And so here's an example, and also what I had to do this comparison is illustrative of the difficulties with the different formats, how these lists aren't very interoperable. So here's the peg list number one. This is the one I just showed a piece of in the previous slide. So this had multiple genes for every locus, but in order to use this table, I had to manually convert it into a table that had one row per gene. And then they hadn't summed the evidence types, so I just added a column and summed up the number of evidence types per genes to get, which is the gene with the most evidence at each locus. Then in the second peg list for the same trait, it was only presented graphically here. So their genes are along the bottom, and the dots mean that the gene had significant evidence of that type. So in this case, I actually did type it into a table. I'm kind of ashamed to say, and then the genes didn't indicate which locus they were at. So I had to compare with a supplementary table to find the locus for each gene. Now, I should have said before that when you're comparing two different studies that are based on two different GWAS, of course you have to find the shared loci, because it's not meaningful to say this, if a locus was found in one study and not another, there's no meaningful comparison. So I found the shared loci and wanted to see how concordant these lists were. So these loci, so these lists had 9 loci in common. And then I asked, how often does the highest ranked gene at a locus from the first list, which ranked multiple genes, how often is that the same as the top gene from the second list? And I found concordance at 6 out of 9 of these loci. I see things are coming in the chat, but I'm almost finished, so let me just finish
got
and then.
Keep going.
Okay. I looked at another example. So this was another 2 lists. I won't show all the details, but there were 22 loci in common, and in this list, only 11 of them were concordant between each other. I think tomorrow Jason is going to talk about a different comparison of some
No.
lists. No?
Not as much.
Not as much? Okay. Well, the point is that there are another couple of lists that we are going to compare, but they each involve hundreds of loci. I don't have the computational skills to do that, and I have a feeling that a lot of biologists probably don't. So I think we should make it easier for them. So my conclusion after doing this survey was the landscape of peg lists is the Wild West. Anything goes. People do everything. They just make it up as they go along. There are a lot of great methods out there, but it's there really isn't any consensus or standardization at this point. So I'm just going to suggest a few minimal standards that came to me while looking at these papers, and we'll talk more tomorrow about standards. Most of these are so obvious, but people don't always do them. So first of all, you need to document the GWAS from which you got the loci with all the standard documentations for GWAS. It's really helpful to specify the boundary coordinates of the loci because those can vary. And of course, describe how each evidence type was generated, what's the reference, what the reference for the method, what is the input data, and how did you decide whether the evidence was significant for each gene? Describe the criteria for that. And if you have a scoring system, you need to document that. I think it really is helpful to present a single peg list and of course to present it format as a downloadable and interoperable spreadsheet or text file without any graphical elements, although it's great to also just include a graphical element to convey to readers your overall conclusions. I do think it's important to present evidence for all the genes at each locus because that is just so much more informative. But if you do that, then it's also important, I think, to have some kind of a scoring system so that it's obvious which gene has the most evidence. Identify the sentinel variant at the locus, use standard nomenclature. That's all pretty obvious too. This last one is not really a standard, but it's an observation that I wanted to make. So a lot of people use pipelines, as I said, but and the pipelines incorporate all different kinds of evidence, but then some people also incorporate that type of evidence separately and end up double counting evidence. So but the biggest example was that I think in 39 papers, people used depict, which does include mouse mutant phenotype annotations, but then they also separately looked at mouse mutant phenotype annotations themselves and counted both those. So
Did you find, Maria, that
worked out
people
for-
were not only using the same evidence types in a duplicitous manner, but they were also finding the same results twice? So for example, if you find that there's evidence for a causal gene for hyperlipidemia, you may find that there's the same evidence for that same causal gene for hypercholesterolemia. And we often find that people double count and make overconfident calls because they're just reanalyzing the same phenotypes.
Right, yeah, I didn't look in that much detail, but that's a really good point, yeah. I think that's it. Yes, that is it. So I have a couple minutes for any questions.
Questions for Maria? You can unmute yourself. Or that's easy for her.
I could make a comment really rather than a question. That was a really great presentation, Maria, and I can totally sympathise with all your pain. So just want to talk a bit about the why the GWAS catalogue is interested in this whole topic, because we used to curate, so we curate GWAS from the published literature, and one of the features that we used to curate was something called that we call the author reported gene. So we have an ensemble mapping pipeline that reports the closest gene. We would also all curate which gene the author said. but we stopped doing it because it was so hard. It was hard for the curators to do. It's very time consuming, and also with no data behind that, it could be. And as you highlighted, Maria, if it's just a figure with dots on it, really, what is that telling us? And I think it's a shame really that journals allow those kind of things to be published with, if you just have a nice figure with no data behind it. So yeah, we stopped curating the reported gene, but we would like to include that information in the GWAS catalog, and that's something we can do if we've got standardized peg lists that we can link to and kind of say, well, this is the predicted gene, the top predicted gene for this variant, but here's all the data behind it. So yeah, thanks for doing that work. And I think, yeah, it was interesting, but to see the proportion that don't provide scoring.
Yeah.
I wondered if people wanted to comment how important they thinks providing a scoring system is.
Yeah, so tomorrow I'll give the results of the survey that we put out to the community that many of you answered. And yeah, I think scoring was popular in the survey. People do think scoring should be included, even though they don't always do it in their list, in their publications.
Hmm.
So how would we, I guess we should talk about this, but like the question to ask, like how would we enforce scoring from a contributor? And how would we develop standards for that?
So in the MR community, it was a lot of work, but we put together what's called strobe guideline. So the strobe community are epidemiologists who have dealt with the exact same problems that are being described by this community for years, reporting standards. And the strobe community puts together checklists of what should be addressed in a case cohort study, what should be addressed in an effector gene study, what should be addressed in a Mendelian randomization study. And then these are usually published in very high impact journals because they get cited by every April. If you can convince the journal editors that they must fill out the strobe checklist for effector gene papers, then the original paper gets cited very ridiculously well. And if you would like to inflate your impact your personal impact factor much higher than it should be.
For sure.
And that's
That's good.
which I think just speaks to the irrelevance of impact factor. But it is a way to conform a community. And the epidemiologists have done this really well. When you try this in MR, and it has at least resulted in some of the Wild West being tamed a little bit, but something that could be considered.
Yeah, we get to talk through those. I've never heard of that. So are you going to mention that in your talk, or can we bring it
I
up
hadn't,
tomorrow?
no, I don't have any slides, but I
Okay,
can.
we can talk about it.
about it.
Cool.
Yeah.
That's great. And then I guess Marina is going to talk a bit more about scoring from the ClinGen perspective. So that'll be really good. Excellent. Any other questions?
Yeah, I did have a question. I alluded to one of my pet peeves is not understanding why the authors picked the phenotype that they did, right? The mouse phenotype or the rare disease, and or even clearly delineating exactly which one they picked. I mean, I think I saw maybe a BMI paper that picked any rare disease that had any body weight phenotype, which was clearly far too broad. Is that something that you found that authors were or weren't clear? I mean, if there's a mouse phenotype column, is it clear how they arrived at that?
Yeah, not always. I mean, that they would, usually it's just there was a mouse phenotype, of interest. Yeah, so that the details are not always provided.
We have another question at EDI.
Yes, it's it's probably the question for tomorrow, not for today, but since we represent the pipeline-based prioritization here, the question is, the presented standards is mostly about the publications. But for example, us, Open Target Genetics, we generate the similar table in systematic manner. We have a lot of features, but we actually can just adjust and create the list of features to comply with the proposed standard. We are not going to publish this paper because the table is actually huge. We can provide with 50 gigabyte or I don't know how big is the table actually, but it's huge to the community. And I simply don't understand how it fits the publication-based standard and pipeline-based standard. So if you see what I mean, there is a problem.
That's like a really
And-
important division. 'Cause
Yeah.
There's a lot
Like,
of,
and we are not only one group who actually produces this type of systematic evidence. Like, I think 3 more groups are actually working on exactly the same thing, and they have probably the same question.
right, and we've gotten, I think that Mira and I went back and forth on this, which is, are these effector gene lists? Or are they the outputs of systematic method applications, right? And I think that it's not clear where one ends and the other begins.
Right, yeah.
And it's probably easier, as you said, like when you have a pipeline that produces it, it's much easier to standardize. Downside is that those often don't match like the kind of internal knowledge that disease communities have, right? And so that's I think where I think Maria, we focused more on for the curation, right?
Right.
And that I think that is a worthwhile distinction as we talk tomorrow, like where those lines are drawn, whether it matters.
Yeah, or figuring out how, based on our different skill sets with open targets, with GWAS catalog and with, the Knowledge Center, Knowledge Portal as curators, how that we can converge those things into an Uber resource, if you will, that could be more useful for the broader community, that does exactly what, you're talking about, Yakov, and what we're thinking about with the sort of the human-centered curation on top of it. So I think that's exactly something we should focus on tomorrow. Other questions for Maria? Yeah, I think we're running out of time.
Yeah,
That's
time to
great.
move
So thank you, Maria. Virtual clapping for all. And I think our next presenter is a suite of presenters, which is great. So these are some great collaborators too. One is which is in the room, one's still in the state, but other side of the state, and another one who's across the pond. We're gonna start with Cassie, who's gonna talk about her experiences. And then I think after that, we move on to Brent and then to Adam. So we have 10-minute talks for each. I'm gonna give a little flexibility. Pop the questions in the chat. We'll keep them if we have to for later. So Cassie, are you good? Ah, there you are. I see you and I'm seeing your screen. Fantastic.
Yeah, can you hear me okay?
Just great. Yes.
Perfect. Hi everyone. I want to thank Maria and Noël for inviting me here today, both to help plan this meeting, but also to present just one method that a student of mine sort of worked out and trying to not necessarily identify the exact effector gene, but how to at least take the list of genes that we're looking at from a GWAS hit and sort of narrow them down. And in doing so, one of my former students, Vishal Sarsani shown here, who's now a computational scientist at the Broad. This is sort of his approach that we worked together to come up with, and we called it GP score. And in essence, what you'll see is it's just a way of trying to combine evidence from multiple approaches together into one sort of prioritization scheme. And we did this using adiponectin as our trait of sort of a proof of concept. For those who don't know, adiponectin is a cytokine secreted almost exclusively by adipose tissue. So we really, in this paper, cared a lot about adiponectin and adipose tissue itself. So as this whole meeting is sort of about, we're trying to identify target or effector genes coming out of GWAS or other variants that are being identified. And one of the things that we considered in this particular paper is the fact that there are all these different methods and approaches that you could use to try to narrow down what the effector gene might be, but a lot of them disagree with each other or have, some are really strong evidence, some are minimal evidence, and we wanted to try to put all of that together into one. But we also wanted to focus on data sets that helped sort of give weight to some things over others. So for example, if we're going to look at eQTL data, we really like the eQTL data that came from adipose tissue because that is the tissue that is far most relevant to adiponectin than many other tissues. So we wanted something that we could sort of customize or give extra weight to that. And so together we built this score where we're integrating a lot of expression QTL data, polygenic enrichment, known regulatory elements, and then also non-functionally informed target annotation and put all of that together. So here are the pieces that we used. So from the non-functionally informed target annotation, we did use distance to the transcription start site of the lead GWAS variant. So in this particular meta-analysis, we had data that was built on multiple builds. You've got HapMap2 imputed variants that are on HG18. You've got really deeply imputed variants, not from TopMed, but from 1,000 Genomes Phase 3. And so we really didn't trust doing credible sets in this particular paper because of the vast missingness across data sets. So we just focused on using the index variant for this. We have transcription start site plus magma. We included CS to G score. And then, as I said, we cared a lot about the GTEx tissue, or the EQTL tissues that mattered most to adiponectin. So adipose tissue is here, as well as some cardiovascular tissues and some other things that we have both subcutaneous and visceral adipose, those sorts of things. We really focused on those in the score, and in the method documentation that exists on GitHub, it talks about how you can select which tissues might be most of interest and how to make sure those are what are included in your data set. For our EQTL colocalization, we use both the colloq approach, but also an LD colocalization approach, because colloq isn't perfect, and so we have evidence from both, along with summary Mendelian randomization, LD score enrichment, and expression modifier scores. From polygenic enrichment, we included some pathways, and expression from POPs and downstreamer, and then for regulatory elements, we have EpiMap and GeneHancer. So we put all this together. This is a sort of easy breakdown of how this score worked. So some of these methods give you p-values for the different genes. Some give you an actual quantitative value. So we wanted to find a way to put all of that together, and there's a lot more detail in the paper if you're really curious for how the math behind this works. But essentially, we're combining the p-values together into one part of the score. That's the PG. We're combining things together that have like a quantitative score together. That's the SG. CS is just a proportion of the support for all 11 strategies. So each gene, we're sort of counting up in a way the number of support across the different strategies, but we're also giving a higher weight to something if it's from adipose or heart tissue in this paper, again, giving it that extra weight for those biologically relevant things. And then the transcription start site is there as well. So just for illustrative purposes, how did it do for adiponectin? So it identified many well-known and novel target genes. So I know this is a little busy, but this is each of the chromosomes that had a GWAS hit from our paper. And then for each, those are the RSIDs or the index variants, and underneath that, it lists the genes that this method prioritized as the potential effector gene for that variant. And so on chromosome 3, we get AdipoQ, which is the gene that encodes the adiponectin protein. Makes sense. On chromosome 16, we get CDH13, which is the receptor that binds adiponectin. And then the number of stars and number of circles is just telling you sort of the percentile that score gave you. So the more stars or circles you see, the better the evidence. Oops. So some other well-known ones, we had IRS1, PDE3A, CCD92. So those gave us some confidence that this is actually working well to identify things that we would have expected to see. Not a gold standard per se, but these genes are all very well known for this trait, with lots of biological evidence to support them. We had many novel loci in this paper, which, when you have novel loci, sometimes it's hard to know what the effector gene would have been. And so here's 4 of the novel ones that we got. And after doing some literature digging, learned more about what those genes are and believed that the prioritization is working. So for example, CSF1 is known to be a mediator of adipose tissue growth. So it makes sense that adiponectin is probably coming from there. ADRB1 is a regulator of human brown adipocyte metabolism. So again, there are these connections to adiponectin. And once you start digging in the literature, it makes sense, but at least we didn't start looking at every gene at every locus. It sort of helped us narrow it down. You can also kind of look at this pictorially. So this is a heat map to help show the amount of evidence for each of the genes in the region. So we did just to be as encompassing as possible, we looked at all genes up and downstream of that index variant for 1.5 megabases, so a 3 megabase window. So these are the genes in that 3 megabase window, or several of them that would fit on the slide, and the amount of evidence. So anything that's more yellow or more of that dark purple is a stronger amount of evidence that we got for that particular gene. And so you can look here across the ones shown in this slide. AdipoQ has the strongest evidence. It has that dark purplish color for the transcription start site, and then that yellow light green for the PG and CS portions of the score. But the other ones do have some level of evidence across those other genes. It's just not as strong as AdipoQ is. And this is just one more example. So here the prioritized gene is PBRM1. Again, this is one I believe is known, but you can see in the locus zoom plot that there's a lot of genes going on in this region. So having to take time to look up every single one of these genes, like from a literature search, would take way too long. And so this was, again, a way of sort of narrowing that down. This is just a circle plot. So each of the lines coming out of the circle is a different variant, and it's showing when we've prioritized a gene, how much of that evidence is coming from which part of the score. So that purple or that red color is distance to transcription start site. You can see for some of these that plays heavily in the weighting of that being a prioritized gene. That tealish color is the summary of all the p-values. The yellow color is a summary of the quantitative scores. When the method uses a quantitative score, and that's purple is just the number of different approaches that prioritize this gene. So you can see some of them that is bigger than others.
Cassie, 2-minute warning, just so you know.
Great, I'm almost done. So we also, I'm gonna run through these 2 slides really quick. We also looked at other traits just because adiponectin was our proof of concept. So we have some FinGen traits. So this one is looking at pulmonary fibrosis and just showing that at this one variant, TERT shows up as the prioritized gene for idiopathic pulmonary fibrosis. And here the EQTL is based on a lot of lung tissues. And then we also have another example looking at thyroglobin gene mutations, or sorry, hypothyroidism, and we looked at the thyroglobulin gene, which showed up as the strongest strongest prioritized gene, which is what you would have expected based on the trait itself. So again, this is sort of the summary of how that score was put together. This is the website for where the GitHub is for if you want to look at the score or use it yourself in the documentation. So again, this was my former grad student Vishal's work, and so I just want to give a shout out to him, as well as Sarah Brotman, who was at UNC and is now at the University of Washington, who helped with a lot of the behind the scenes work as well.
Awesome, well done. Thank you, Cassie. Virtual clapping for Cassie as well. So, and Cassie has, how many, what, we have a list that's already in the portal for that as well?
Adiponectin list, yeah.
Yeah, that list is in the portal and accessible, so, you know, we've been able to take what you did and make it accessible, but that's also taught us some things. Those are some things we can talk through tomorrow too, as well. Awesome, without further ado, any burning questions for Cassie? I know there's some questions in the chat, we're gonna answer them, but they're more global ones, I think, and I think we missed them live within the talk, so we'll circle back to them tomorrow. So with that, shall we move on to Brent? Yeah?
Sure.
Brent, you all ready? All right, Brent's in the
Which
room.
audio do I use? Do I turn
Just
on my?
keep this, yes.
As long as I can see your screen, I can see your screen, you're already there.
Yeah, that looks great.
And if anyone does any trouble, just make sure you guys can hear us. Brent, why don't you speak, just like, test your voice to make sure they can hear you.
I'm hoping you can hear me.
So a little muted, it's a little muted.
Okay, maybe I'll talk a little louder. Is that better, Eric?
Yes.
That's
Okay.
better.
All right.
Great, thank you, everybody. Thank you kindly for the invitation to come and talk. It's wonderful to be in Boston and see some old friends and work on a problem that's very important to all of us. Okay, so I'm gonna start off. I do have a conflict of interest in that we have spun a company out of our lab called 5' Sciences that uses human genetics and other omics to identify and validate drug targets. I'm going to talk about a study that we did just to frame a bunch of problems in the field. And so this is, as other people have presented, a method called the effector index, which aims to identify the probability of causality at GWAS loci by assigning a score of 0 to 1 to every gene below us. So it really, what we tried to understand is, would this method recover positive control genes? And that's what I'm going to spend most of my time talking about, positive control genes, and really applaud the work that Eric and others have done to try and sort of field, because as Maria pointed out, in just the reporting of the predicted effector gene list, it's a wild west. But I think in actually what are the effector genes or the positive controls is an even wilder west. And I'm going to show you some examples of that. So to try and sort out this problem, we started naively, instead of looking at thousands of traits and diseases, we really wanted to look at a few, but really try to understand. And understand their problem. Because if you start off with a few, when you expand to many, hopefully you've identified some of the problems before you go out to thousands of traits and diseases. These are the ones that we looked at in our original work on this paper. They were selected because we could find very, very large GWASs for them, and we sort of understood to some degree some of the puzzle. But of course, something was missing, and still not sure what it was. The thing that was missing was the positive control genes. So when taking a first bite out of something, I always try to be small and try to be as clear as possible. We wanted to really focus on Mendelian disease and targets of approved drugs, but then that leads to millions of other questions, which I'm going to go over in a second. So to do for that list, we came up with a reasonably large number of OMIM genes and about 113 drug targets. And I'm going to really focus on the drug targets for a second to highlight some problems. So I happen to practice medicine, and I happen to be lucky to have a lot of students who practice medicine. And sometimes you can be taught a little bit about what are the medicines that you should use to treat disease. And you can use some of that prior knowledge to try to understand if the actual drug targets that are advertised in a bioinformatic catalog or database are actually true. And many of you know that in the world of clinical medicine, physicians just follow guidelines. That's all I do. That's all most physicians do. So you can actually look at the guidelines to see what are the actual guideline approved drugs and the targets. It's a really painful job, but you can do it with enough people in enough time. And so this really leads, I think, to what Maria was pointing out, not just like the PEG problem, but the EG problem or the egg problem, is that the effector gene list is pretty, I think, flimsy, depending on how you source your data and what you want to look at. So let's just take through one example. I could have picked many papers, but I just pulled this out this weekend to take a look. I just did a search of the inflammatory bowel disease drug targets and open targets. Okay, so that would be a reasonable place to download your data if you're going to do an analysis. And there are, I don't know exactly how many, but there's about 35 genes there. And then you can just use your clinical knowledge or your knowledge in the field and try to come up with, are any of these sensible? So this is what you get. You get a lot of non-causal or non-specific genes. So let me just take you through a few examples. You get the drug target for fentanyl. So what would be the probability of you doing a GWAS for inflammatory bowel disease and identifying as a causal mechanism that leads the human to get inflammatory bowel disease, the drug target for fentanyl? I would suggest it's 0.
Wow.
We all know that vitamin D does not treat ulcerative colitis or Crohn's disease. I wish it did, and I've spent a lot of my life trying to show it does, but it doesn't. Then you have non-specific. So azathioprine and mesalamine, these are good drugs for, as well as methotrexate for inflammatory bowel disease. But we almost have no idea how these drugs work. They work in very broad mechanistic systems. And if you dive into them, you can end up with a whole bunch of targets. So if you look at multiple sclerosis, this is another fun one to look for. You get the whole KCN family. About 25 genes are causal drug targets for multiple sclerosis, just because there's one drug which is rarely used that's a potassium channel inhibitor. And you get all the potassium channels because that drug has off-target effects on about 35 different KCN genes. Then you get to midazolam. So midazolam is a medicine that you use to put people to sleep right before their scope. So you scope these ulcerative colitis people all the time, and you give them midazolam so they don't remember. If you're over the age of 50 and you've had your colonoscopy yet, you probably got midazolam. You're never going to find that in a GWAS of ulcerative colitis. Caffeine is a hit.
That's awesome.
I wish it were. I've had 3 cups of coffee every day. Lidocaine is a drug target for ulcerative colitis. Why? Because, well, you need to give people lidocaine before they have their colonoscopy. Dextromethorphan, an anti-coughing medicine, is a target for ulcerative colitis. So this is just, you could do this on most diseases. You just spin through these and be like, oh my goodness, this is what we're trying to find. So how is it that we could find this? Because we can't, because these do not either cause disease or are so non-specific that we're hitting like hundreds of targets related to those drugs, or the drugs are full, we have no idea what they're doing. So that's really the problem that we tried to address in our paper, clean it up for just a small number of targets. But I applaud people like Eric who've tried to clean this up at scale across a domain. But this is really a field-wide problem, and I'm really happy that the folks here at the Broad are taking some leadership in these trials that are trying to fix this problem. Another Wild West is Mendelian randomizations that I talked about. Maybe some of you have seen that there's like everybody who's graduated from high school, I think now has a Mendelian randomization paper published on PubMed. My children have several, and the problem is that the standards of just reporting are so loose. Many years ago, this actually, and I wouldn't suggest this to the community unless people have the time. This took us years to put together this this actual strobe MR list. It is being used. It hasn't cleaned up the field, but at least has cleaned up the reporting of the field so that people can understand what it's doing. And it's really required by many of the leading journals now for that publishment of the randomization factors. So something to think about. So I'm just going to switch back to the effector, and I'm going to show you some of the work that we've done and how the results can change if we just switch out those positive controlled gene lists, because they can change importantly. So we used a bunch of features, and I'm not going to get into that. There were locus-specific features as well, and people have already been over that. We used a whole bunch of different models. We ended up using XGBoost, but that's not the point that I'm trying to make. I'll show you the results. I'm going to point out really nice work that was done by Mark and Anuba that actually manually went into the type 2 diabetes loci and tried to say what are most likely to be causal. And I'll show you what happens when we switch those expertly curated positive control gene lists. Okay, so how does the effector index perform? And I'm going to show only precision recall curves. Hopefully everybody is somewhat familiar with these. So I want you to just remember a number here. So this is just for those 12 traits and diseases. The number is 0.24. Okay, it's pretty low. But that was when we published this, I don't know, 3 or 4 years ago. So if we just at that time then switched out to this positively or expertly curated list, which at that time included GWAS, then just for type 2 diabetes, the effector index moved from 0.35 to 0.65. So that gave me a lot of hope that as we got larger and larger GWAS studies, that we could actually find the causal genes. Reminder, if you did an GWAS study for ulcerative colitis, would you hope to find the drug target for fentanyl? Of course not. And so that is a way that we could overcome that without bringing in a lot of the bias of the prior literature. This is just a slide showing, as Eric pointed out, that you really have to try to beat locus level features, and you should also very clearly show in these sorts of papers how does the nearest gene perform, because of course we need an algorithm for that. And I'm going to show you now what happens in work that we've done inside 5', which is trying to develop a next generation of the effector index, which we call PrimeX, how it performs when we change the positive control gene list. So every algorithm published in the literature seems to beat every algorithm published in the literature. And the problem with that is because I don't think people are being dishonest, but they're developing and evaluating their algorithms on different sets of outcomes, right? And when you compare them, then you magically do better than anybody else who didn't use your same trading data. And so this is just a nice example of this. So if you use the training data that PrimeX was used to develop in version 1, you get a very high area under the precision recall curve of 0.75, and it compares very well to open targets. But this is biased, and it's biased because of the fact that this is the data on which Lai Jiang, who developed this method, trained it on, right? So you really should be switching this out and trying to use the actual targets that we used for training the other algorithms, which brings us to Maria's point, is that it's often really hard to know what were the positive control genes that were used in the papers that are published. We spent a lot of time trying to dig through papers to find those, and it's really hard to find. I'm just going to see what happens when we use actual other positive controls. So you can see, as expected, a decrease in performance if we use GWAS as our positive controls. This just compares to open targets. But you can see that the area under the precision recall curve used to be about 0.21, and by improving the algorithm, we're looking at 0.59. You change the positive controls to the open targets list of positive controls, the CIDR, which was used in Rondeau's very nice paper that was published in 2024 in Nature Genetics. You can see, again, an important improvement from 0.21. When you just don't use the CIDR, but you actually use the ones that we believe were used in the open space paper, although we had a hard time understanding, you do see important improvements in the algorithms. Metrics depend upon which positive controls were used. And really, I think, in my humble opinion, the best that could come out of a dedicated group of people such as yourselves is to try to come up with a list of positive controls that people could use to compare the difference of their offering. Other problems is that when you only use a limited set of diseases, its applicability to other diseases are knowing, but at least you can start to test what are the problems when I do this. And then the last thing that I would point out humbly to the field is that the accuracy estimates that everybody are reporting are deflated because most of the causal genes are not known. And this will actually improve. So,
Brent, can you go back? What do you mean by that? Can you explain that accuracy estimates are deflated because most?
yeah, so let's say, let me just give an example. So let's just use ulcerative colitis because we have, because we talked about that.
Yeah.
So you can clean up the list that you get from a database like Open Targets to get rid of what are clearly non-positive genes. You can use different thresholds for inclusion of those genes in different scoring systems. But at the end of the day, I believe the vast majority of causal genes are actually not known. So if you take like a BMI, GWAS of more than 500 independent loci, we don't know the causal genes of most of those. If we even knew 250 of them, and we knew that 50% of the causal genes in every GWAS, then the number of loci upon which we could train our algorithm would increase importantly. We don't have that.
Got it.
So I think it's deflated, and it will improve with time as long as we update the positive control genes.
Right. Okay.
Okay. So summary, I think this field has a lot of promise, and I've invested a lot of time and people in our lab and company have. But using really just a simple set of metrics that are available for most GWAS, we can generate reasonably helpful probabilities of causality across many traits and diseases. And if we can build more field credibility by standardizing what we do, then it will just be more helpful to the people that we're trying to help. Mm-hmm. Importantly, prior biological information is sometimes used, but I really don't think it's required, and performance is improving rapidly. So welcome any feedback and suggestions. And I'll just thank people who work on the effector index. There's lots of people around the world who work very hard, and we actually got some funding from the FNIH to work on this many years ago. And we're very thankful to the work that's been done at AMP to be able to show the results, and also thank the folks who worked at Pfizer to put this together, and thank you for your time.
Thanks, Brent. Virtual applause again. I hope that bothers you right now. Any questions for Brent?
I guess just a comment. I really liked working on that work with you, and then I like the approach of you have a positive control, and then you can weight the different metrics based on your positive controls. I get really frustrated with papers and others that say we don't know which metric is going to be best, so we're just going to throw them all in and add them all up. But then, right, you can always go back. I mean, there are the well-known genes, or the genes that you expect to come up, and you should check them. So I really like the rigorous approach you took there, and I think that should be applied more often.
Beautiful.
So maybe in those rock curves you were showing, the negative examples, were those all genes not in your list, or those at GWAS loci not in your list?
So it just depends on what the positive control
Looking for
sets
XLOS, for
are.
examples. Is that?
Yeah, so you'll never find the fentanyl or targeted cholesterol. So it's like I can stake I'll bet you my house. If we get
Sure.
sample sizes up to billions of people, we'll never get that.
To define a negative, is that does not have an XWAS signal, or at a GWAS locus and does not have an XWAS signal?
I'm not sure I understand the question.
Well, you have a positive label,
Yeah.
which is going to be, it has an XWAS signal, right?
Mm-hmm.
Then the zeros, the negatives,
Yeah.
it can either be anything that doesn't have an XWAS signal, or it could be, it has to be in a GWAS locus.
Oh,
without the
okay.
XWAS
Yeah.
signal.
So that's a good point. So that's a very important point to underline. So I believe, and it'd be interesting to see what other people have what they would do. I believe that you should only be interrogating loci in your confusion matrix which have GWAS signals, because those are the only ones that you can find, because you are only starting with GWAS data.
Right.
You can't find you can't find a causal gene using any of GWAS locus to gene mapping methods if there's no GWAS signal. And so we've extracted those from all of
Got it.
the,
Okay, so-
from all of the confusion matrices. So I don't know what would be a good example for that, but they get extracted.
All right. I think we should move on to our next speaker, and that will be Adam. Adam, we've also had the joy of working with in constructing and presenting some of his work for coronary artery disease, and he's actually going to talk about that experience and others, I'm hoping. Yes, he is. Awesome. Adam, we can see your screen.
Good. Hopefully the camera will pan round and show me as well in a minute. So when it zooms in, there we go. It's it's
hey,
working
you
very well.
are.
Yeah,
Yay.
good.
I was
Good.
there
I'll leave-
first.
Good. Well, I'm glad you're pleased with what I said I'm going to talk about. My favorite example of causal gene mapping is actually from our metabolite work. We've done many of these kind of QTL studies in proteomes and metabolomes. For example, this one where we identified just over 400 kind of credible sets, I guess you could call them. And we managed to assign causal genes for about 2/3 of these. And actually, this is my favorite method of kind of causal gene annotation because it's actually send the list to Eric, and Eric will send back a beautifully annotated set of evidence-based statements, involving papers going back decades, kind of showing why this is the right gene for the enzyme. And I guess this speaks slightly to my question earlier about, should we have different ways of defining causal genes based on different types of phenotypes, which is something we talked about a bit in the break here, because I think you can do this very well for something like metabolites. But then thinking about complex disease, this is work that we did in coronary artery disease led by Krishna Aragam at the Broad and Tao Jiang, a PhD student of mine, where we discovered about more than 200 different regions of the genome from our GWAS. You then have the question, well, we kind of wanted to go beyond this kind of, let's pick a few examples and scan the literature. Let's come up with something that's a bit more evidence-based. It's a bit more objective, hopefully. It's something we can transparently report, and we can kind of come up with our predicted effector gene list. But of course, something like coronary artery disease, predominantly people having heart attacks or other manifestations that are similar, many different tissues, many different risk factors. We can't just dive straight into adipose or straight into liver. There could be many different routes, which we know about already from the handful of genes that we do know. This is the schematic from our paper, where essentially the approach we came up with was a kind of, I guess, a collective kind of consortium approach developed over many Zoom calls, but 8 different predictors, and I'll take you through them kind of one by one in the subsequent slides. But essentially 8 different ways of coming up, taking these 200 or so regions, looking, annotating the genes in the regions against each of these kind of metrics. And then coming to the question that we've just been touching on really a bit about kind of, how do you score these? Do you weight these? We took a very simplistic approach, which was adding them up and kind of telling you, kind of picking out the winners. So we do give you all of the information in the paper and supplementary tables, but then kind of saying, well, whichever gene wins, i.e. has the most number of supporting predictors out of those 8, is the most likely one for that region. And you can see on the right here, you can kind of just draw a simple schematic of that, and you can kind of see there are a few with 7, PCSK9 and LDL receptor, kind of really well-known ones come out at the top, which is nice. But you can kind of work all the way down. And so what this looks like overall is we said there were about maybe 220 kind of causal genes we could prioritize, but you see this heavy skew on the numbers of concordant predictors. So, if you wanted to set a slightly higher bar, for example, and say, well, you need at least 3 concordant predictors, then that number instantly drops to more like 100 or so likely causal genes. Just to maybe touch on some things we discussed, we did discuss several times, should we come up with some kind of score or weighting scheme? People wanted to kind of use some sort of machine learning type method. A number of reasons we didn't do that. One was kind of we're only looking here at one phenotype, and we have a lack of positive control genes to really kind of test and train models on. Secondly, some of the information is quite sparse, as we'll see. So some of it, definitely, I think, kind of subjectively, we might all say, well, this is a stronger predictor than this, but how should we How should we weight this? And thirdly, I think we wanted something that was kind of simplistic to understand. So we didn't want to kind of come up with a number that people then couldn't instantly reverse engineer and kind of see where we'd got to. So I think we kind of accepted that this was simple and pragmatic. No doubt we could do better with a more complex system, but for one single disease, this is the way we were going to go. Quite a few papers seem to have cited ours and used the same kind of image, but just to say that we didn't dream this up entirely ourselves. And I guess this shows that effector gene lists and systematic evidence go back some way. So I think we consciously or subconsciously pretty much took this from Robert Plenge and co.'s rheumatoid arthritis GWAS back in 2014. So this is certainly been going on for some time. We weren't the first to invent this approach. So most of these are quite straightforward to understand, the types of evidence. Just to give you some illustration from the numbers here that we presented in each of these kind of blue discs, we said, well, we don't necessarily know. We can't tell you for all of these regions whether we've got the right answer, but what we can tell you is whether each of our predictors, how many of the regions it's giving you a prediction for. So in this case, 220 for the nearest gene because it's ubiquitous. And then the first number, the numerator here, is the number of those where it agreed with the overall answer. So we said, let's assume that we've got the right answer by integrating these 8 predictors. How often does this particular predictor get you the right answer? So you see nearest gene here agreed with the answer 84%. Of course, this is slightly biased because it's including information from that predictor. But we thought this at least told you about the kind of agreement of each with the winner. So as well as nearest gene, we also used whether there was some kind of coding or important protein altering variant as well. I guess partly based on the kind of, problem with GWAS is they're mostly non-coding variants that you're finding, but actually where you do get a coding, a missense variant or something more important, then this is probably a good predictor. And again, you can see that actually about 50 of these, we did have a protein altering variant. So it's quite a useful predictor in terms of the quantity. And again, about 80% of the time, it was agreeing with the overall winner. Rare variant associations we thought were important as well. Again, this kind of standpoint that they're easier to identify the causal gene. So where we overlap, where we have a GWAS signal that's in the same region as a rare variant signal, it's probably telling you something useful. This one's more tricky in terms of how you do this. We have GWAS catalog, but we don't really maybe have as good data sources on rare variant analyses. So it's a bit more of a literature search in some way. We included exome array studies, whole exome sequencing studies. Again, there's different thresholds used, so it's a bit difficult to, it's a bit more woolly how you define exactly what's a gene that came out from this. So, and this was much sparser, as you see, so only 10 of the regions here did we actually have some information. So again, this might be one where you would say, well, we could outweight this, but actually it's not maybe going to be that useful for a predictor in aggregate, but it will do well in certain regions. Similarly for monogenic disorders, I kind of said there's a rare disease here that's relevant. Key here being what's relevant, particularly for cardiovascular. There were a lot of different diseases that we had. We had a lot of debate about including or excluding certain diseases. I think this will be one of the things you can do well with a single phenotype. But of course, the challenge here, if you want to develop a predictor that works across universal phenotypes, is how you're going to assign, how you're going to tweak that to make it well predicting across lots of different diseases and traits, because it was pretty difficult to do with one. And I suspect here, you see here, we only got 69% concordance. I suspect that's because we're a bit liberal with some of the diseases we included. EQTL we thought was useful, but I think only in restricted sense. So a bit like Cassie described earlier, we restricted to sets of tissues that we thought were most relevant. Again, this is kind of tricky to do with something like GTEx, where there's a very broad range of tissues, but we also included a data set called StarNet, which is patients with coronary artery disease who only have this kind of very specific set of tissues. So we took those, we took GTEx, which instantly raised the problem of how to combine those. So this is just a screenshot from one of our supplementary tables, but we quite often found that you might find evidence from StarNet that you didn't see in GTEx, even where the tissue was there and the tissue was well powered. So particularly for liver, perhaps. And we never really got to the bottom of whether this is because it's patients with the right disease, whether it's because these are living people rather than post-mortem, whether it's because certain tissues are better in other data sets compared to GTEx, whether it's statistical power, etc. But we certainly thought there was value in including more of them. And we just counted any eQTL here from either of these databases. Brent mentioned about drug trials, kind of approved drugs and Mendelian randomization. We thought both of those were useful sources because we trusted those. But again, pretty the drugs is reasonably databaseable, as Brent showed, but kind of MR was a bit literature searching and a bit subjective investigator led, I would say. So, but for 17 of these regions, so again, not widely useful across the whole set, but probably quite predictive when you do find a result. Mouse knockouts has been touched on several times again, and particularly as people said it might be useful for cardiometabolic. We thought it was, and again, there's very standardized information as Eric showed earlier, like the MGI database or the IMPC database. Again, I think the challenge here is how to restrict to a set of phenotypes that you think are capturing diseases or phenotypes that are relevant to your disease, but are not so ubiquitous. So, you know, we had growth and body weight here, for example, so probably some false negatives in there as well. We use POPs, which I won't go into the methodology too much. It's been touched on a couple of times. Again, this one's really useful though in the sense of coverage. 220, you know, you get results for every region. But I think this also touches on something else we've discussed, which is when you don't get a kind of yes/no binary answer, but you get a score from these, how do you incorporate that score? Do you just take the top score? Do you take anything above an absolute score? Do you somehow rank this into your system? One thing I think thanks, nearly finished. One thing I think is important, and we thought was important, was how to validate this. How do you know that your algorithm's doing well? You can compare against the nearest gene, but you ideally want some way of ground truth. The way we did this is we decided collectively to try and come up with a gold standard set of known causal genes before we designed the algorithm to kind of validate and tweak the this methodology on. So there's a set of genes that we sort of agreed. Of course, there were disagreements. There was bias towards people's own experimental studies. So maybe a different crowd of people might have not come up with exactly the same list, but there was broad consensus. And 25 of these 30, the single highest ranked gene in the locus was what the right gene. 3 of them, we had joint winners, so we couldn't separate 2 genes. And 2 of them were, if you like, wrong. We picked up the wrong one. One of them, I think, because there may be multiple causal genes in the region. And the other one, the endothelin one, probably because of distance. So that's probably an example we think that's greater than 500 kb away. As Eric showed earlier, they can be as far as a megabase away. And actually, several of our predictors were too narrow. So we probably weren't looking as far away as we should have done. I'll skip over that in the interest of time. And that one. And maybe just finish with some thoughts then on kind of what we learned from the process. Certainly thought when looking at one disease that tailoring for that disease was helpful. So picking up maybe having data from patients, but particularly from cells and tissues and those kind of conditions in the mice and the monogenic disorders was useful. One thing we didn't do that we thought about that I think could help here is actually looking at related diseases and traits. So given that LDL cholesterol, blood pressure, diabetes are all risk factors for coronary artery disease, if we've got known causal genes for those, and these intersect and co-localize, then maybe we should kind of pull through that information. So some kind of multi-trait, multi-disease weighting could be useful. I think there's value in having multiple data sets because they provide complementary and orthogonal information. So even within the same predictor, like GTEx and StarNet, but then of course there are challenges about how to integrate within those, which I think is one better than the other. Do you just sum across them all? Gold standard sets we've touched on, how do we define these, how do we best validate the methods, but I think that might be something we should catalog or have in terms of reporting standards. Have you shown using some kind of set of genes that this works or this is better? And I think in our strive to maybe kind of come up with objective and systematic methods, we have to acknowledge we largely ignore the literature. So a lot of the studies that people were citing that were evidence for our gold standard genes were, the single paper that dived into a GWAS locus and kind of proved to beyond our collective doubt that this was the likely causal gene, but that doesn't get picked up by the data, but the kind of data sources we were using, that was almost the PubMed bit. And I think somebody touched on earlier, there's kind of guilt by association there as well. So I think kind of linking together, most of this was one by one region at a time, but actually linking those together probably could have improved our prediction as well. So I think there's still additional predictors and methods that could be used here. And I'll finish there.
Thank you, Adam. Virtual clapping and API can do in person there. Thank you. All right, what we're gonna do is because we have 45 minutes left, we want to respect the fact that we want to try to adjourn right around our scheduled time. I think we're gonna flip-flop the discussion for the end, so that we make sure we have time for Marina. That we can save your questions and save our discussion for them, because I think we're all chatting offline here too with the questions we're gonna pull together so we can have a discussion. But I think in order to make sure we give respect to our last speaker, we'll make sure she has time. So Marina kindly agreed to go a little earlier. So why don't we shift to that? And then we can discuss at the last, however many minutes we have. Hopefully we'll have about 15 minutes at the end to talk. So Marina, do you want to share your screen?
Sure.
This is a totally different perspective. I'm very excited that she's willing to be here. It brings in the rare disease, what ClinGen has done in this space to sort of give us a sense of how that framework for standardization was done for ClinGen and how we might apply that to what we're doing in common disease.
Great. Can you guys see my slides and hear me okay?
Yeah.
Perfect. Okay, so yes, changing directions entirely. It's been really interesting to hear the discussion today, and it reminds me a lot of some of the beginning ClinGen standardization discussion. I've been with ClinGen for the last 8.5 years, and ClinGen has really existed for the last 10 or so years. And so we really spent a lot of that time, developing standards, developing frameworks, and getting buy-in, because you can have as many standards as you want, but if no one used them, what's the point?
What's the point?
So I will tell you about a couple of the approaches here. So I'll give you a brief overview of ClinGen so you can see what we're working with here. And then I'm going to give you a couple different case examples because we heard cats all day. I threw a fun picture in because you guys are like a fun, sassy group of good picture users, so like that. We heard cats every day. But I'm going to talk to you about 3 different case examples. So standardizing terminology, and this will be in an example of a Delphi survey. Engaging disease experts, and this is the example of gene curation framework standardization, where we came with a point scoring system and people widely use it now. And then also piloting at scale, engaging the community. And this is our variant curation framework standardization. Okay, so the Clinical Genome Resource, or ClinGen, was funded primarily by NHGRI in 2013. The current funding is through 2026. It has 3 separate grants based at Baylor and Stanford. I'm part of the Broad Institute and Geisinger grants, and then UNC and collaborators. It's governed by a steering committee and includes all the PIs. I'm one of the PIs, and additional key representatives from each grant, as well as NHGRI and ClinVar, which we have a very close partnership with. It was designated as a global core biodata resource a couple of years ago, and as of August 2024, we have over 40 working groups, over 120 expert panels, and over 2,500 individuals from 69 different countries that contribute to ClinGen. So our mission is to build and support authoritative central resources that define the clinical relevance of genes and variants for use in precision medicine and research. And I will say this largely started as highly penetrant monogenic disease, but we are exploring more complex disease now, polygenic risk scores, and things in that arena, as we know it's quite relevant. So this is our infographic that people may have seen before. And really, we need to first start by aggregating genomic and health data from a variety of different sources: patients, clinicians, laboratories, researchers. And then we take this data and we curate it based on ClinGen's critical questions. Is a gene related to disease? Is a variant causing disease, variant pathogenicity? Is a change in an amount of a certain region or gene going to cause disease? And how actionable is a condition if it's found in a screening setting? And then these groups of experts that are largely volunteer-based, so international experts, we assemble them in different disease areas. They pretty much volunteer their time. We have a small group of paid staff off of all of the grants, but they curate And then we disseminate this expert curated knowledge entirely publicly. Of course, we publish it in papers too, but largely we pride ourselves on publishing our curations to our website for use by the public as soon as they're done, really with the goal of improving patient care through genomic medicine. So I'm going to start with my first example of a Delphi survey and harmonizing terms. So ClinGen is large itself, but then it's also part of other international consortia. And one of them is the Gene Curation Coalition, or the GenCC. And this is a larger organization that includes ClinGen and all of these organizations you see here, a variety of publicly available resources and also private clinical testing labs, which we felt was critical. So we all came together for gene disease validity as a gene causing disease in a monogenic, highly penetrant situation context to understand the approaches and classification systems of these different efforts, and then to develop consistent terminology for a variety of different things. So gene disease strengths, inheritance, allelic requirement, and mechanism of disease, really to get on the same page so that we could post gene curations from any group willing to share. So the GenCC database itself is like a ClinVar, but it's for gene curations. It's assertions of gene disease relationships. And then once we had this database, we were going to mine it and use it to resolve differences and collaborate on gene curation projects. And so before we could even do this, we had to compare our approaches because we were all sitting in our own silos and talking about things, and we might have been using the same words, but we realized we all meant different things. And so what we did is we took a pilot set of gene curations and we all curated them. And then we talked about it. Why were they the same? Why were they different? And you can see that there's kind of a gradient where where green meant like high level of evidence, and red meant low level of evidence, and yellow orange was somewhere in the middle. And so you could see based on the type of resource, there was a gradient of like strength of evidence, but it's because we were talking about different things. And so this pilot was very helpful for us to understand what each resource did, because even though we used each other's resources, we didn't curate within each other's resources. So this was very helpful to us. And so it just identified some reasons for the discordance. We were maybe talking about different definitions of disease, high versus low. We were maybe curating for different reasons. Should something be on a gene panel, or should we just be curating the gene disease relationship entirely without thinking about like what panel it should be on for testing? And some genes have claims for multiple diseases, as you know, and maybe we were just talking apples and oranges. We were curating different diseases. Or, as you know, the literature changes very rapidly and we use the literature. And so maybe something was just a little bit older than something else. So really understanding these differences helped us to focus on resolving them and harmonizing our terms, because we needed to start with the same definitions to then eventually get to the same terms. And so we compared our terms and they were a mess. They were all over the place. We actually had opposite people were using the same terms to mean opposite things. So one group was using red to mean a strong gene disease relationship, and another one was using red to mean a weak gene disease relationship. So of course, when we were talking, we were very confused and things were all over the place. And it really just showed that we needed to harmonize these terms. So we performed a Delphi survey, and for this process, it had multiple phases. And so the first part that we had to do for this survey is to harmonize definitions. So we all needed to agree that we were talking about the same thing. And so we wrote definitions for the strength of gene disease relationships in monogenic highly penetrant disease. And we needed to make sure that these didn't contain any of the proposed terms that we were going to survey the community about for the names of all of these buckets. So for the next phase, we prioritized. And for a Delphi survey, with each round, at least in the beginning, you suggest every single term that you could possibly think of. So everyone suggested all the terms that they used and any other terms that might be within the realm of possibility. And these suggestions were solicited from all GenCC members. And then once we had all of these terms, we used a modified Likert scale to express our level of agreement, disagreement, or neutrality for suggested terms. And we felt that The neutrality part was also quite important because, as you know, working together often comes with compromise, and you need to understand if someone feels neutral about something versus they cannot live with something, as far as, you know, when you're trying to come up with terms. And so that's why we felt it was really important to hear if someone really liked a term, they really disliked a term, or they could potentially live with it so that we could come to common ground. So during the survey, the Gen CC members expressed their level of agreement, disagreement, and neutrality, and then they were prompted to add additional terms for the next round of the Delphi survey. And so then for phase 3, we did iterative surveys to refine the term list. And for these, really the terms that scored the worst were removed in each iteration until we got to a much more concentrated level of terms. So for the second set of surveys, we extended this to Gen CC members and their curation staff. And so after the survey, terms with scores greater than 2 standard deviations below the mean. And so we used positive +1 for like agree, +2 for highly agree, and negative numbers for disagree. So like -1 for disagree, -2 for strongly disagree, and 0 for neutral, and summed them all across all of the terms. And so at that point, we removed terms with scores that were greater than 2 standard deviations below the mean. And then we gave this to the international genetics community. And so we tried to send this to a bunch of different genetic societies, along with an optional 10-minute explanatory video to say why we were doing this and the importance of this, so that people really had background as to why we were doing this survey. And so we got 241 responses. And then in phase 4, we did consensus and implementation. So survey 2, which was the Gen CC members and the curation staff, and survey 3, which was the international genetics community, had 93% term agreement, suggesting that our choices are in agreement with the larger genetics community. So we took these terms and they are used to standardize submissions to the Gen CC database, with the additional category of supportive for less granular submissions by some curators. So this was a way that we are now able to display thousands of curations on our website using the same terms, and actually some, I don't know if yes. So we have 22,000 curations that are displayed using these terms now. And actually many submitters in the beginning only mapped to these terms, but many of the submitters now have gone back and changed entirely to these terms using our harmonized definitions. So that's the first example of standardizing terminology. So I'm going to go to the next one, which is a semi-quantitative framework of engaging the experts. So we developed a semi-quantitative framework to classify the strength of evidence for the role of genes in disease. This is the publication in American Journal of Human Genetics in 2017, and it's scoring genetic evidence and experimental evidence, largely from the published literature, to come to these classification terms, which are the ones that we actually harmonized in the Gen CC that you saw before. So this happened a little bit before the Gen CC example. And so for this one, in order to develop this semi-quantitative framework that people would actually use, and we had to kind of, you know, find a systematic way to test the semi-quantitative framework and make sure it made sense. And so first, we developed it. You know, we have a genetic evidence matrix, we have an experimental evidence matrix, and we kind of talked through like what numbers of points do we think relative to other points would make sense, but then we needed to test it. So for this publication, we chose 33 gene disease pairs across a range of different diseases. Once again, we're talking about highly monogenic, highly penetrant disorders for this. And then we had dual curation. So we had 2 curators because we really wanted to make sure that this was a systematic way to curate gene disease relationships, because before it was just the Wild West. Like, you know, people would say, yeah, this gene was associated with disease because we found some variants in a couple people who have this rare disease. Therefore, it's associated, this gene's associated with disease. So we had 2 curators independently take a gene disease relationship, take the published evidence. We needed to make sure that they were on the same page so that they had the same amount of evidence, so we could compare appropriately and score. And then most importantly, I would say at the end, is we would then have these curators go and have a conference with an expert in that disease area and say, we did this curation, this is what our scoring got us to. With your expert gestalt, like, do you think that this makes sense? Is this framework behaving how you would expect it to? And then we measured the concordance. And after some tweaking, we were actually pretty happy that the concordance between curators who didn't talk at all but had the same amount of evidence, they had the same publications, was 94%. And then the concordance between curators and experts, which you would expect would be a little bit less because experts, you know, truly know the field, whereas curators are just using a framework, was 88%. But the genes that differed really only differed by one strength level, and we do have a bit of flexibility built into the framework. And I will show you that here. So this is kind of like the description of the points here. We have 12 points of genetic evidence, 6 points of experimental evidence with 18 points total, and then asking the question of replication over time. And so you'll see we built a little bit of flexibility into this framework because we have international groups of experts who use these frameworks, and we feel that they really should tailor the frameworks a bit into their disease areas. So you'll see that there's a little bit of a points gap in between each strength level here. So 1 to 6 is limited, whereas 7 to 11 is moderate, knowing that you will end up with 6.5s. And then you need to talk to your experts in a disease area and decide, you know, should this be a limited gene? Should this be a moderate gene? So great, you have your framework here, but you need to get some buy-in. Like, are people going to use your framework? This point scoring can take quite a bit of time, and ClinGen curations do take quite a bit of time. So, you know, what's the impetus for using this scoring framework? Well, there are a couple things. First of all, we try to do as many curations as possible, and then we put all of our evidence on our website, including all of the point scoring by the international groups of experts as to why they upgraded, downgraded, or scored something, so that it can be used by the genetics community. Second of all, we engaged professional societies. And so there's actually a policy statement now that the American College of Medical Genetics has a technical standard that says anything with a ClinGen curation of moderate or above should be included on diagnostic panels. Therefore, this kind of, you know, gives everyone the carrot of either look at ClinGen's curations or perform a curation consistent with ClinGen standards so that you can put something on a panel. Whereas anything that's strong or definitive should really be used for predictive tests. You should have much stronger evidence there. So not only did we develop this semi-quantitative framework, we made sure that the right professional societies were also on board with this and could make policy statements consistent with that. And that's definitely brought a lot of buy-in. So how is this used in everyday work? Like, are people uptaking our frameworks here? So this is a select kind of sexy example from one of the GSEPs where we had a Brugada syndrome expert panel. And a lot of times the testing panels in the past were really just kind of like a McDonald's meal, like more genes, bigger value, which is not necessarily the best, because then you just end up with all these uncertain significance variants because there's not legitimate gene disease relationships. And so they went and they looked at all of these genes that were on panels for Brugada syndrome, which is an arrhythmia syndrome. And they found that only one of them had valid gene disease evidence. And so this is a screenshot from Invitae where they removed all of those genes from their testing panel and just included one. Of course, they still had the add-on for the other genes if a clinician wanted to choose that. But they entirely deleted all of the other genes from their panel based on ClinGen's curations. Okay, last example, engage the community. The community is always willing to be a part of your work. And, you know, ClinGen is extremely community engaged. We would not function without our rare disease community because they are all our volunteers. So we have our variant classification framework. And this framework, I think I have This framework has been iterated on for years now. So the very first one was in 2002, and the most recent one that we've been using is from 2015. And in this 2015 one, there were harmonized terms. And so these are the terms that you've seen that are benign, likely benign, uncertain significance, likely pathogenic and pathogenic. But before that, people were using all kinds of terms, pathologic, mutation, disease causing, those types of things. And so first, before we did any updates to these guidelines, we did a survey of, assessing adoption of the guidelines. We put out this framework for classifying variants in a highly penetrant monogenic disease, but is anyone using it? So we sent it to 195 US-based and 170 international laboratories registered in the genetic testing registry. We got responses from 65 labs, and 95% of these labs reported using the guidelines. So really, once you have a framework, you can't just, pat yourself on the back, give it to everyone, go right off into the sunset. You need to check in and see if your framework's adopted and useful. So it is, people are using it, but you need to find points of improvement too. And so when we ask people, how they're using the guidelines, 25.8% of US labs implemented a more advanced version of the guidelines that added caveats for usage and different strength levels of evidence. And this speaks to something. This means that we were maybe a bit too broad and a bit too generic in these guidelines. Like, yes, we gave people standards, but we need to keep giving people standards, because this can lead to interpretation differences downstream if advanced implementation is different between the labs. So this really meant like we still have more work to do in the variant curation space. So we engaged even more professional societies to get buy-in for standards. For the last set of standards in 2015, it was the American College of Medical Genetics and the American Society for Molecular Pathologists. We added even more people this time. So ClinGen is involved this time, and the College of American Pathologists. So really, you need to engage as many relevant professional societies to get buy-in for standards as possible, and international societies if possible too. And so this is just a photo of all of the members of that working group that are working on these new guidelines. And you need to harmonize adjacent frameworks. That's another thing that I wanted to show again in this iteration of the guidelines. So for this one right here, you'll see it's important to have standards for sequence variants, but they should also be harmonized with standards for copy number variants, because they were kind of, iterating on themselves independently and in parallel. And based on the type of sequencing technology, you're going to have crossover between the two. So this might not be exactly what you would be thinking of in your area, but you need to think of things that are parallel but might be overlapping or adjacent when you're setting frameworks, because you don't want people in a slightly related, like adjacent field, doing something completely conflicting with what you were doing. So for example, there's a lot of singular points-based system here, whereas in the copy number variants, they were using decimals. And so it was really hard to understand, the scoring in sequence variants versus the scoring in copy number variants. And so they came together to this points-based system. They used harmonized terms because they were using different terms before. So that's just something to think about too when you're developing your standards. Continuously engage the community. So these draft rules were workshopped or presented at all relevant professional meetings for the last 2 years. And each time these draft frameworks were presented with audience polling, which was used for decision making. So this is an example where they have a suggestion in their current draft, but they actually use audience polling to ask the audience what would be the most appropriate point value for a particular situation. And there were at least 5 to 10 questions every time. And this was, factored in the iteration of the guidelines. Have a public pilot of the rules. Invested users will help. So the most recent version of the variant curation guidelines, this group has a sign-up option for clinical labs to volunteer to pilot the new guidelines and provide feedback. They already have 100 labs across the world who are volunteering to test these guidelines. That we've developed in ClinGen a lightweight pilot calculator that'll save and export the classifications so they can be emailed to the standard setting group along with some comments. And when a draft is finalized, it still goes to the members of each society for comments and feedback. So most importantly, provide a mechanism for updates to your framework, version it, be willing to receive feedback about when it works and when it doesn't work. And sometimes it's frustrating because you've just done all of this implementation, you've just done all of this, maybe website or curation interface development, and someone comes back and says, this really doesn't work in this one tiny piece. And you want to just dig in your heels and say, no, this is my framework and this is how it is. But I think that that's the most important thing that I've learned is the community really is there to help you make it better. And you need to be nimble and flexible when you're developing guidelines, because that's the way to the best version of the guidelines. Science is always evolving. Why shouldn't guidelines? With, some bit of standardization, of course. So in conclusion, standard setting requires objective and transparent metrics. Delphi survey, measuring concordance between curators when you're curating for gene disease relationships. Engagement of the community, even patients or the public if a standard relates to them. We have some, disease naming advisory rules, and we have engaged the patient communities for those, because a patient's disease or a disease name can be part of a patient community identity. So think about who are your invested users. And then flexibility. If something in a standard doesn't work, it should be updated and improved. So I just want to thank everyone in ClinGen. I am one of many, many members of ClinGen, and I couldn't do any of that without them and the funding sources there. And if you have any questions, my email is here. And with that, I will stop.
Thank you, Marina. That was great. All right, we finished with some time to talk, but questions directly from Marina. I have a question. It's more about a community question. So the GWAS community, the disease community around common disease is very much, different disease areas have their own way of doing things, and they're in their little community. And, it's a common disease, so they're really good. They are pretty good about making their, their GWAS statistics accessible. They have good ways of working together. But I would say it's more of like community-based, these little vignettes across common disease. In the rare disease space, do you feel like because of and I almost feel like there's a lot of people working in the common disease space. It's like common, lots of people, different ideas, not a lot of synergy except in small communities. What's the community like in the rare disease space? And do you think that the rare disease community is more open to these type of thing than the common disease space? Or what are your thoughts on that? Just gonna gauge you.
Yeah, it's interesting. I would say that the rare disease community is much like you just said for the common disease community. I work across maybe like 15 or so different expert panels. And one I can think of is my expert panels in like craniofacial malformations and skeletal dysplasias. I feel like that disease area has existed since like dysmorphology has existed. And so if a person looked different, they named the disease something different. And that was way before genetic testing had happened. And so to harmonize, with ClinGen standards was a bit of a struggle. But I would say as we were developing standards, for example, for disease naming, we made sure to go to every single one of these communities and ask them, like, how are you naming diseases? And what are you doing within your community? So we could kind of try to come to like a middle ground. But yes, I would say that everyone maybe has their own challenges. And that's why we built a bit of flexibility into the framework and said, like, these things are deal breakers. These things you should do with the standards of your community. And so there are certain things that are deal breakers. And then the other things that we're trying to develop standards, like we brought everyone in and brought them all to the table and said, like, what are the things that are most important to you to retain within your area, just to try to like come to a middle ground.
So I have an interesting question, and there's another question in the chat, but like, do you feel like folks in the rare disease space are more passionate and directly focused on solving the disease, whereas in the common disease space with these effector gene lists, you find a combination of people who are trying to understand the disease, but also people who are computational biologists or thinking about like cool methods or like I want to take this approach. And so they have a different skin in the game, perhaps. And you think that made it easier for adaptation, like adoption, excuse me, of those standards to your community?
I think there's maybe we're a little clinician heavy, but we do have researchers. We have people who are doing MAVES, a lot of like functional assays, people who are interested in like the display of functional data. And so I would say that's researchers and biological questions have become a little bit more of a percentage of ClinGen than they were in the past, when it was much more clinician driven and laboratory and clinician heavy. So I would say, we're heading that way. We're heading in that direction.
Cool. Question for the chat is from Carl, says, do you envision Gen CC ultimately replacing all of these resources that went into making it? All the resources that went into making it?
It might be different in 10 years from now, but I would say that I think the strength of Gen CC is still linking to the primary data sources that have like a lot more granular data than the Gen CC itself. So I don't envision it replacing any of these resources. I think it's really a good place to aggregate all the resources together, and then you would go to the resources to get the primary data. But I could be wrong in 10 or 15 years. As of now, we definitely don't have the development capability to aggregate all of that really granular information that all of these resources display so well.
Other questions, different sites or on Zoom from Marina before we go into broader discussion? All right, Marina, that was excellent. That gave us a totally different perspective, and I think we have a lot we have to chase. As there's an Oscar speech that Matthew McConaughey gave once about like you have to have somebody to chase. So we need to chase the standards by what ClinGen is doing for the field within our field. Okay, so with that, let's pivot. Do we have a slide to kind of guide some discussion, Maria?
Yeah,
Perhaps?
I made a slide with some
Beautiful.
broader questions.
Instant.
And I think for this, we should encourage people to just unmute and talk maybe, because rather than having to read the chat, that might be better.
Yeah.
And so these are just guidelines. Anything goes.
So I think this morning we heard a lot about folks who are building lists, and I think about different evidence sources in terms of how they use them, right? So we thought about how Eric approaches finding the causal gene and his approaches from Cassie and Brent and Adam. And I think that helps us understand the landscape really well. But what we want to think about, particularly as we pivot to tomorrow, is how do you make those all that work accessible to others? And therefore, by doing that, you have to develop standards for them to pull that information into a centralized framework or structure that you can make it accessible. And so I guess some things to think about is, as you all have made these lists accessible, how have you done that? Like, one of the things that, many of you in the room have made the list accessible through the portal, but like, let's talk about that experience. What did you need to do there? What would have been helpful when you were creating and making a list accessible? Like, did you find that it's actually easier for me to just make a figure and just put it in the back of my table and forget about it? Like, is that actually the way you want to go? Have your list been used by others? That's a question that actually I want to prompt. Like, have you actually found that the work that you've done, either any of you or folks who have not spoken but have generated lists, have you found someone actually used your list to do something else? And what did they do? Why did that matter? Because I think that teaches us about the standards we want to impose. And what's the purpose? And ironically, do you know if any of the genes in your list have been experimentally validated or used as a target? Also information that's useful to us, because this teaches us how we might need to develop standards, what type of metadata to capture, what type of evidence sources to link out to or to incorporate directly. And then do you plan to share your list? Like, what's the plan for a lot of you? These lists were generated some time ago. Are they living entities? Are they going to go on? Do you plan to iterate them? How might that work? Because this also, again, all feeds into developing standards. And then the great question that Maria put in here is, which of the ClinGen approaches would be useful for our PEG community? We don't have to start with this question necessarily, but I thought that would be a really good way to bring us all back to what we're really trying to do tomorrow, which is develop those standards. So that'll open the floor for comments, questions, statements.
Yeah, we've got a comment here in the room at EBI.
Beautiful.
Yes, so before we start the discussion, I wanted to return to the question I asked in my presentation in the very beginning. So what is actually the effector gene list? And as far as I see, there is like a consensus that effector gene list is basically all genes in the region around, like in the locus or around the sentient variant with the genetic evidence. So it's not simply the list, it's actually a table. Because like we didn't actually agree on this very simple concept yet.
What do folks think about that? It's a good question.
Yes, what is the effector gene list? Like,
Yeah, I mean, I agree with what Yakov is saying. So we're calling it a list, but it's simply a list of genes is not what we mean. We mean a table of evidence.
so but it's table for all, like the evidence for all genes in the region, or like
So I think that is a question for the discussion tomorrow, but I think that's certainly what, from the point of view of the organisers, that's what we were proposing, that it should include all genes in a region. Is that, would you agree with that, Noël, Maria?
Yeah.
Plus the score attributed to that gene,
Yes.
right?
but is it only protein coding genes, or is it like all genes in the region?
These are all topics for tomorrow, I think.
Yeah. So when you mean by region, yes, so I'm actually just capturing these, so this is great to iterate that, that's useful at the time we have. So you're saying like, is it all genes in the region, or is it all genes, but goes into be any of the genes that the contributor decides they want in their list, and all the evidence sources with that? Actually, like the list of genes may not just be defined by the region, it's defined by the entire set of results from GWAS or something.
Yeah, I mean, my take would be this would be best to defer till tomorrow. when
I'm
we
just trying to
get
capture the
the-
actual question,
Yeah,
yes.
so the exact definition of what the list
Yeah.
is, what features it needs to have, the universe over which those are defined, all of those things are what we either, I think as a community would either say we should define that ourselves, or an alternative approach could be that we encourage the generator of a list to define them.
Right.
And those are all up for discussion, I think.
Just to add a bit more questions for tomorrow's discussion. So again, what should be this list focused around? Around one GWAS or around the publication? Because like some methods are actually focused on analyzing several studies together. So it's like you can't actually say which study is specifically used for this particular effector gene list. So like, just as a question to discuss.
That's a good one.
One of the things that I caught was, I think Cassie and Adam each mentioned overweighting EQTL in a tissue of relevance. Now, I'm assuming for Cassie, it was adipose, and Adam, perhaps heart for your CAD studies. But I think number one is, well, tissue of relevance assumes that we know what tissue is relevant
Right,
for a particular
we
phenotype.
don't always.
And of course, it also implies that, and these are probably cis-EQTL that we're studying. To what extent are cis-EQTL preserved across tissue in response to a local variant? And it, of course, it raises the issue of circulating monocytes that are often used because they're easy, right? So I think trying to get a handle around that would be important.
I like that.
I was just trying to get a handle on what were the positive controls that were used in the study, where else would be helpful.
So making sure that that's incorporated in the deposition of the list, or the representation of the list, okay, I like that.
Noel, I think your third bullet point there captures nicely the discussion that we were just having relating to ClinGen, which is, I think there may be a difference perhaps with the GWAS community to the rare disease community, in the sense that the users, the generators may not always be the users here. And I think we spend a lot of time kind of as people doing GWAS coming up with lists, but not necessarily always so connected to how it's going to be used. And I think thinking a bit more about what the uses are, who's using
Right.
these, how are they using them, what do they want to see, because that may be, you know, sometimes it may be the people in your community who are also doing experimental studies, right? So they want a shortlist or a high confidence gene for somebody to go off and start their PhD project on because it's got a high likelihood. Other times it might be a pharma company, right, who wants a big list to pick from and things that actually fit into there. And I think perhaps end users might have quite different use cases and therefore different things they want to know or want to see. And I think we often, as the people writing the GWAS papers, often do that in kind of sort of ignorance of this, in a kind of, I've gone beyond my GWAS paper that has 3 new hits where I can just kind of look them up in PubMed and write a bit in the discussion about it. I need to do something more systematic, but it's really from a publication point of view, which I think is what you were coming back to. Is it from a helping get my publication out or and giving readers some context, or is it actually for downstream use cases? And I think we're very much moving towards those downstream use cases, helped by your portal and other more agnostic initiatives like Open Targets. But we need to kind of, I think we should make sure we're trying to include the end users here as well, because that may not just be the people who are the generators.
That you're talking my language. I totally agree with you. Because the end users, I mean, we've seen some of the outputs of that with what we built so far, simply from like way back to, I mean, one of the lists was actually used in an RFA that the NIDDK put out. So this matters to the community. It's like a 5-year project that 2 awards ensued from a heuristic that was done for diabetes, which is an awesome expression of how important this is to like experimental biology and to other resources. So like it matters to like funders even. So it's like an incredibly there's an end user that thinks beyond just, you know, I'm gonna put my results out and use an experimental all the way. You also have users who are gonna take those lists and have their postdoc just kind of pull them all together and kind of like look for overlap and do it in a very like non-structured way so that they can take top 10 genes and make CRISPR knockouts. I mean, we have an example of that, and it actually led to a paper from Anna Gloin where that was done. So like that's a real thing. And those end users are the type of people we want to, you know, really make sure that we enable them to make this more systematic or easy for them to use. So I think that's a great point. Other points to sort of lay down for tomorrow. You're going to hear from the GWAS catalog tomorrow a little more about their experience with developing standards for the GWAS community for GWAS summary statistics, which I think will really help us think about the as you said, Adam, people who are generating the list often are folks who are in the human genetics space, who aren't necessarily thinking about an end user as much as like they need to make the results acceptable for publication, you know, for funding purposes. But we want to pivot that focus to like, how is that useful to others? How might others view those lists? So we're going to hear about that tomorrow. Maria's also going to talk about her experience with actually what we've presented so far. Like so we have about 170 of these lists accessible on the knowledge portal right now. And we actually have them, like if you search a gene on the portal, you actually get this gene has appeared in these effector lists. And I don't know, that might be helpful all by itself, but you have to make that more structured and synthesizable. And that's, you know, we're just at the beginning. Other thoughts over at Open Targets and EBI that we want to make sure we lay down for tomorrow, Laura and others?
I was just typing in the chat actually. I wondered if we could ask like is there someone on the call who'd like to share an example of when they've used a peg list in their own research? Because I definitely when we did the survey, there were people who said, yes, I'm a consumer of these lists.
Yeah, that's great. Anybody on the chat, right? I mean, on the Zoom right now that wants to mention that, so we make sure we address that tomorrow more specifically.
We've done that.
Yeah, good. So we should definitely make sure that that's addressed. I want to know what you did and how that was useful and what you did to do that.
And I can see in the room here, Yakov's packing up, so I think he's keen to go. But just to follow up on your question, so would you say that you're talking about combining lists together, or combining studies together rather?
Well, it depends. So it's like if we are talking about open target genetic perspective, we actually provide both. And for us, it's like different level of evidence.
So when you're talking about combining studies, would you have already done the work to predict effector genes based on one study and then combine that result with results from another study.
Yes.
Okay,
Yeah, so it's different levels.
yeah,
So it's like you can have this prediction score on the level of the study, and you have this prediction score on the level of the disease. And actually, we can generalize it even more because, again, as Eric Fauman already presented, that basically one particular variant or prediction set can be associated with different diseases. And if it's the same variant or the same prediction set, it probably supposed to lead the same prediction, but currently we are actually analyzing them separately, and it can lead to a situation when credible set is basically the same, but just because of the different evidence we have, we can predict it differently. So something to discuss.
yes. So to me, that's like, it's like a two-step process to get the standards for presenting a study-based prediction. And then if you have all the sufficient metadata from all the studies, then you can combine them or compare them to get to the next level that you're talking about.
Yeah.
Yeah.
All right, I'm mindful we are 1 minute over, but that's pretty good, all things considered. I think that, so I really appreciate that. We packed a lot in. We had some actually really good discussion. I think it's our job as organizers to sort of synthesize this and sort of bring the questions back to us tomorrow. Your all job is to I'm thankful you're all here. Come back tomorrow, hopefully, because that's actually where the action is gonna happen. We're gonna have the same Zoom tomorrow, is that correct, Mackenzie? No, it's different.
It's a different Zoom
Different
link,
Zoom.
so you will have gotten the invitation. It's also in the group meeting notes document that it's a different Zoom link, and also it'll be a different meeting room for the people here.
Yeah, and
Beautiful.
just a reminder, even though the invite says 8:30, that's because we wanted to give some time for people to join. The event starts at 9.
We'll start at 9, yep. And so everything from today's session will be in that Google Doc for the presentations and things like that. And those active notes, if you think of anything at night or like this afternoon, if you want to put it in the Google Doc so we can make sure we record it for tomorrow, we will do that. Please do that. Maria, any other comments?
Yeah, last thing I want to say is that there are questions in the chat that we haven't addressed. We are capturing the chat and we'll put it into the Google Doc, and so everyone can contribute to the discussion about all those questions.
All right, with that, I want to thank our speakers. I want to thank our organizers for making it through day one. I'm looking forward to tomorrow, and I hope to see all of your faces or your little names on the Zoom box tomorrow. So thank you again, and we'll talk to you again.
Thanks, everyone.
Thanks so much. Thanks, everyone.